Good morning, everybody. Mandy, can you hear me?
I sure can.
Okay. Well, good morning, everybody, and welcome to the 2021 FAPSI Virtual Research Symposium. Last couple of years, we've been knocked out of the park here. 2 years ago because of the ice storms, and last year because of the pandemic that initiated. So, and each year we were trying to get our keynote speaker to give a presentation. I'm glad that he stuck in there, and 3 times is a charm. The 3rd time is a charm. So Jason Reicks, our keynote speaker today from Florida Food Products. Thank you very much for sticking with us. I just want to mention that our agenda today is going to be, we're going to start out with graduate student presentations. We have 13 of them. That'll be followed by our keynote speaker, Jason Reicks, and then followed by 7 undergraduate presentations and 2 faculty presentations. So, and then later we will have an awards presentation as well for the awards. Just for the sake of being precise, we really have to stick to our time limit of 10 minutes max. So hopefully the students can get their presentations in under 8 minutes to allow a couple of minutes for questions. If not, I apologize ahead of time, we will unceremoniously stop you at 10 minutes because we have to get on to the next speaker. Okay, so and each presenter's responsibility to initiate the screen share for their presentation. Okay, we can't do that. It has to be done by you. So hopefully you know how to do that by now. We've had a year of Zooming under our belt, so hopefully everybody is adept at that. Again, I'd like to thank our committee, Mandy Gross, Karen Smith, Chuck Willoughby, myself. We spent enough time in rehashing all the details of this symposium during the course of the oncoming year. Mandy also with all the website and the registrations and so forth, and Karen Smith with a lot of the other details. And Chuck is going to be, he's in the background too with the kind of gathering all the judges and so forth. And Chuck, do you have anything to say in terms of sponsors or any other thing that I might have left out here? You're on mute. Okay.
There we go. Yes, I want to say thank you to our sponsors, the Stillwater Centennial Rotary Club, as well as the Oklahoma section for the Institute of Food Technologists, which is a co-sponsor of this, as well as a help sponsor awards for our participants. And then also want to thank the Oklahoma section for the International Association of Food Protection. And because of their support, we're able to have this event, even virtually. It's wonderful. And then also because we are not doing poster presentations this year, we've gone all virtual. The prizes have been doubled. So we've got $500 for first place in the graduate category, as $500 for the first place in the undergrad, and so on. So, and of course, at the end of the day, we'll announce who that is, how much they won, and tell them the check's in the mail. I'm back to you, Pete.
Okay, well, with that, we still have a couple of minutes. I'd like to appreciate and thank everybody for attending here. You know, this has been 3 years in the making. I guess we had the 2 years of delays, so we're glad to. Do we have our first speaker? Are we ready at all, or should we just wait to stay on time?
Let's go ahead and wait to stay on time.
Okay.
Maybe start just a couple minutes earlier, and that way, any of the students that have classes who are trying to target their specific time aren't late.
Okay.
So,
well our one of the reasons that I'll just add that one of the reasons we went after Jason Reicks with Florida Foods with natural curing of meats is that some of the research that I was involved with, we utilized natural nitrite, which is a product that they produce. And so I thought it was kind of nice to have them present in regards to what is the significance of this product to the industry and so forth. And I'll give some of our research data at the very end. I think I'm the last presenter today. So I'll give some of that, I'll include some of that data in my presentation on the use of natural nitrites in processed meats. And I see Melissa's there. Morning, Melissa. If you may, I would say to the presenters, I don't have all your correlating associated departmental associations with me in front of me. So if you may introduce yourself after I introduce you as to what your department or your professor may be, you have that information readily. So you can provide that as part of your startup. So with that, I think are you ready to go, Melissa?
Let me just add, Pete, that as the presenters are going, you've noted the 10-minute mark. If we start getting close to that, I'll give everyone a verbal warning, a 2-minute warning. And then that way they have an idea they need to wrap up or that we're heading towards the last question.
Yeah, we would rather finish up before the 10 minutes so that there's a little bit of time for questions. But if not, we will have to stop at 10 minutes to get the next student, the next presentation going. So apologize if it seems abrupt if it comes to that, but you can understand. We're trying to stay within our time frame. So, and we have a fair amount. We have, let's see, 13 graduate students, 7 undergraduates, 2 faculty. So that's pretty good for a Zoom presentation. That's about as good as we normally have, isn't it, Mandy? Okay.
Yeah, pretty good.
I know it's nicer to have this in person. We're starting to get, being proficient, and it's easy to get these Zoom things going. But again, it doesn't compare to the in-person networking and camaraderie that you get when you're all there face to face and in person. So we're getting close. Okay.
I think now's a good time to start, Pete.
Okay. Well, Melissa, are you ready?
I think so.
Okay.
Can everybody hear me?
Oh, yes.
Okay, great. Let me make sure I can share my screen. Okay, can everybody see my screen?
Yes, we can.
You can see my slides. It looks like grubs, cooked grubs.
I thought so.
Okay, perfect. All right. Okay. Well, let's get started then. Good morning. My name is Melissa Reed, and the title of my presentation is Student Views of Insect Consumption, a Q Methodology Study. I'm a PhD student and a graduate teaching assistant for the Department of Entomology and Plant Pathology here at Oklahoma State University. And in one of the courses that I'm a teaching assistant for, the students are offered the opportunity to consume insects for extra credit points at the end of the semester. And every semester, without fail, 3 groups of students emerge. The first groups of students are not going to eat insects. No way, no how, and it doesn't matter if they need the extra credit points or not. The second group of students have no problem eating insects, and they march right up and quickly consume enough insects to receive the maximum number of extra credit points. And finally, the last group, which consists of the majority of students in the class. These students really, really need the extra credit points, but they feel pretty squeamish about eating insects. So seeing these three distinct groups of students form semester after semester made me wonder, what factors influence a student's willingness to consume insects? And what could possibly be done to sway students, like the students in the third group, to eat insects, other than offering them bonus points, of course? So before I get into the methods of this study, I'd like to give you a little bit of background information about insects as a human food source. So due both to the small amount of land required to keep insects and the small amount of feed required to farm them, insects are a much more energy efficient source of animal protein than traditional Western choices such as cows, chickens, pigs, and fish. Additionally, in order to accommodate the ever-increasing world population, the UN has recommended the addition of insects to supplement human food sources. Currently, insects supplement the diet of 2 billion people worldwide, mainly in Asia and Africa, and approximately 2,000 species are currently consumed by humans. And while eating insects seems to be a rational solution to the challenge of efficiently supplying animal protein to our growing world population, many people, especially those in the Western Hemisphere, are unwilling to even contemplate eating insects. Therefore, the purpose of this study was to identify, explore, and classify the factors regarding insect consumption by college students at Oklahoma State University. So now that you have a little bit of background information about insects as a food source, I'll briefly go over the methods I used in this study. This study explores the perspectives of insect consumption by employing Q methodology, an approach to studying subjectivity in the population. And the first step in a Q methodology study is the development of the concourse. The concourse is essentially a collection of statements about the topic you're researching. We created our concourse from prior research, interviews, and the author's knowledge of the research topic. And step 2 is the selection of a Q set. Using the statements that were generated during the development of the concourse, we established 3 key categories concerning an individual's willingness to consume insects. And then using these 3 categories, we were able to narrow down the number of statements to 41. Step 3 is the selection of a P set. For the P set, we recruited 29 participants from the student body at Oklahoma State University. These students ranged in age from 18 to 44, were comprised of 17 males and 12 females, and ranged from freshman to PhD with a variety of majors. Step 4 is the sorting of statements. Statements were numbered and typed on small rectangular pieces of paper. And then based off of the prompt, what are your thoughts about eating insects? Participants pre-sorted the statements into 3 piles: statements that are like me, statements that are not like me, and statements that they feel neutral about. Participants then refined their arrangement of the 41 statements by assigning each statement to a ranked column within a 41-cell gridded form board. And finally, step 5 is the factor analysis using the software PQ method. This software identifies patterns and emergent themes in data. So now that I'm finished going over the methods, I'll discuss the results of my study. The results of a Q study generate factors. Each factor represents how a respondent with a correlation coefficient of 1 with a particular factor, otherwise known as an idealized sort, would have ranked ordered the statements. These idealized sorts, like the example shown on this slide, are used as a basis of interpretation from which a description of each factor is produced. This factor analysis identified 2 major factors, which we will call factor 1 and factor 2. Data was analyzed using these 2 factors in a principal component analysis, or PCA. Defining sorts, as denoted by an X in the table, were chosen as loadings reached a significance of 0.40. There were 20 defining sorts for factor 1, which are highlighted in green, and 6 defining sorts for factor 2, which were highlighted in gold. And then we had 3 confounding sorts, which were highlighted in blue. And a confounding sort just means that these participants' Q sorts possessed a significant factor loading for both factors 1 and 2. And in this type of study, confounded sorts cannot be used in the construction of the factor estimates and therefore must be removed from the study. So based off our results, we generated themes for each of the factors. And the theme for factor 1 sorts is that insect consumption is good for me and good for the planet. Factor 1 sorters believe that insects are a solution to feeding the world and that eating insects promotes environmental sustainability. They also believe that insects are a good source of protein and vitamins and are safe to eat. Moral or religious beliefs do not deter Factor 1 sorters from eating insects. And the theme for Factor 2 sorters is that insect consumption is repulsive. Factor 2 sorters believe that eating insects is disgusting and weird, and that eating insects is acceptable if they are in a survival situation. This group is afraid that eating insects will make them physically ill. So in conclusion, my study provides the deeper understanding of student perceptions of insect consumption. I found that the students I surveyed had two major perspectives when it came to insect consumption. Factor 2 sorters believe that insect consumption is repulsive and therefore had little interest in consuming insects. It is my belief that there's likely no strategy that can be implemented to get students with this perspective to consume insects. Interestingly, though, the majority of students defined as Factor 1, and what influenced these sorters is that insects are nutritious and that consuming them is good for the planet. Surprisingly, Factor 1 sorters' perspectives were not influenced by trends, family or social pressure to eat insects, celebrities promoting insect consumption or insect recipes, nor high-end restaurants serving insect dishes. Therefore, we believe promoting Factor 1 perspectives that insect consumption is good for me and good for the planet is likely the best strategy when introducing insect consumption to university students in cultures where insects are not traditional food sources. This concludes my presentation of student views of insect consumption at Q Methodology Study. Thank you for your time this morning, and I'd love to answer any questions you have.
2 minutes for questions.
This is Nurhan Dunford. I have a question.
Hi.
You talked about environmental sustainability. I was
Yes.
wondering if there is any study, life cycle analysis on the insect as food, human food. I'm wondering about the carbon footprint of using insects as food as compared to animal-based proteins or plant-based proteins.
There has been studies. Most of the insects that they use for food are crickets, and they can keep them in very small areas, and they don't require a lot of food and water as compared to, like, if you think about like a cattle feedlot, how much area that takes up and how much food and water those livestock consume. But I don't have the data with me.
Thank you.
You're welcome.
Hi, Pete Mariana here. As a food microbiologist, I'm kind of curious, are there any diseases or food safety concerns that should be, concerned about in regard to consumption of insects or insect byproducts?
Yes, I mean, some insects are safe to eat, and then some aren't. I mean, we wouldn't want to consume cockroaches because they do carry diseases, and so you wouldn't want to just go out and pick up insects off the ground and eat them. Definitely, you'd want food grade quality insects, ones that were deemed safe to eat. Now, in other countries, I don't know, I'm sure that they go out and collect insects, but and they sell food grade quality insects. They're just expensive. And that was one of the questions on my survey was, if they were more easily available and more, because that's my problem with eating insects is the price. They're just so expensive. Yeah, I mean, there's food safety issues, I think, with any food that you eat.
This is Kanika Bhargav.
We're out of 10 minutes.
Okay, well, thank you very much, Melissa, for that presentation. Our next presentation is by Gustavo Rosas. I hope I pronounced that right. Effective cherry pulp on rheological properties and quality parameters of dough and batters. So with that, Gustavo, it's all yours.
Thank you very much. Hi, good morning to everyone. I'm glad to be here. I'm Gustavo Armando Rosa Sanchez. It's quite a bit long. But I was in Oklahoma State University 2 years ago, developing this research, and I'm glad to say that I was working. Well, actually, I'm working with Dr. Sorba Hernandez and with the Dr. Patricia Reyes Duarte. Uh, I'm from the Veracruz, Mexico, and like my home, if I can say that, is Tecnológico Nacional de México here in Veracruz, Mexico. So thank you for this opportunity. I'm glad to share our project, effects of coffee cherry pulp on biological properties and quality parameters of dough and batters. Let's say. Okay, first of all, the coffee cherry pulp is the first and most abundant byproduct from coffee production. According to the International Coffee Organization, coffee world production in 2019 was about 10.5 million tons. Uh, this byproduct is about 50% of the world fruit. So that means a display of 5.2 million tons of coffee byproducts in 2018. The coffee cherry pulp is rich in dietary fiber. I'm sorry, it's rich in dietary fiber, amino acids, and polyphenol compounds. On the other hand, bakery products are widely consumed globally, which are mainly made with refined flours that are deficient in dietary fiber, among other nutrients. So coffee cherry pulp could be used as a potential ingredient to fortify bakery products. So our objectives in this project was to determine the effects of coffee cherry pulp powder on the rheological and baking quality properties of muffins, batters, and bread dough. The caffeinated coffee cherry pulp was obtained from Grupo Techver from Veracruz, Mexico, and the commercial spring wheat flour was donated by Shawnee Milling Company from Oklahoma, United States. As we can see in this table, the coffee cherry pulp powder is higher in dietary fiber than the wheat flour. So for this work, we use the coffee cherry pulp powder with a particle size less than 150 microns, 3 levels of substitution for bread dough, and 3 levels for muffin batters. To analyze the effect of coffee cherry pulp powder, we perform the creep recovery tests and a frequency sweep test, the bread making method, and the crumb firmness procedure. The only difference was for muffins that there is no standard for baking. So we use a commercial recipe and a AB standard procedure instead the ACC. So one of the essential factors in producing leavened products is the viscoelasticity of dough and batters. The creep recovery and frequency sweep tests are formal rheological methods to analyze viscoelasticity. And we correlated the rheological results with baking quality parameters like volume and crumb firmness. These are the creep recovery curves plotted with the average of Kelvin-Boyd model parameters for gluten in the figure A and for dough in the figure B. The figures shows a significant decrease in creep compliance in gluten and dough when the coffee cherry pulp powder substitution level increase. This means that both systems become more rigid. Samples with higher creep compliance values at 100 pascals of stress have higher deformation. Lower values of creep compliance were observed in the dough than gluten because non-gluten components provide strength to dough, as we can see in this figure. And we observed that the curves of the control and the first level of substitution overlapped for gluten. And that can be explained because the fraction of coffee cherry pulp is removed during gluten isolation step. These results show that both matrix systems were affected in their viscoelastic balance. Then we have the results for sweep frequency test for muffin batters. For tan delta, we observed a significant decreased tendency, which means that the storage modulus values were higher than the loss modulus value. In the next graph, we can see the difference in increase the frequency test moduli as the level of substitution of coffee cherry pulp increases. For example, we can see a greater distance between modules in the last level of substitution than the other treatments. When the substitution of coffee cherry pulp increased, muffin batter showed more solid-like behavior. This can be explained by a greater interaction of hydroxyl groups, decreasing water plus desired effect in the system. And finally, for the bread and muffins quality, we study firmness and volume. It is interesting to observe a significant increase in the breadcrumb's firmness, much higher than the muffins, 147% for bread and just at 34% for muffins. The bread and muffins volume presented a decreasing tendency, but in bread was since the first level of substitution, and for muffin, just this difference is just in the last level of substitution. So the crumb firmness increased with a substitution of coffee cherry pulp powder, and the volume in both systems decreased due to the dilution of the protein network and the weakening of the gluten matrix formation, as the rheological test showed when they was correlated with the baking quality parameters. For the last thing, the coffee cherry pulp is a good source of fiber, but has a lot of negative effects in both systems. So the substitution of wheat flour, by coffee cherry pulp powder increases the stiffness of dough and gluten and increase the solid-like behavior in muffin batter. This is a first approach that has been performed without any additive. So more research should be done using dough and bread quality and bread additives like emulsifiers or enzymes. We try to valorize the coffee cherry pulp as an ingredient for bakery product. And we believe that this is a way to add nutritional value and reduce the waste and the environmental impact. And the Burger's model parameters that we use were good predictors for bread quality. As the compliance were lower, we had lower volume for bread. And on the other hand, there's no too much negative effects for the muffins. And that means that this product can be more suitable to be substituted with a coffee cherry pulp powder, because also the chocolate flavor mask the greasy, earthy taste flavor that the coffee cherry pulp has. So thank you very much for your attention. That's all. I'm
2
glad
minutes
to
for
answer.
questions.
This is Nurhan Danford again. I was wondering why you're concerned about the environmental impact. In your abstract, you said severe environmental impact. This is a natural product. Is there any toxic compounds in the material that you're concerned of?
Yes, there's been some studies that the caffeine of the coffee cherry pulp react in a way with the metals of the soil. So the presence of the coffee cherry pulp also decrease the pH of the ground, and that has a lot of environmental impact in around and river or water contamination and soil contamination. Here in Mexico, there's about just in one state, like 400 municipalities that produce coffee, and they are small producers, so they don't have the technology to use or the knowledge to use the coffee cherry pulp, and so they just throw it away. So they just put it in a place and they're stay till they get rotten, and that cause severe environmental impact.
Thank you.
Yeah, you're welcome.
I just got one last question. Gustavo, are you saying you're actually presenting from Mexico?
Yes.
All right, we have an international presenter. We can say that we are international in scope. Okay, thank you.
Thank you very much.
Okay, well, thank you for that. Our next presenter is Morgan Denzer, and Morgan will be presenting on dark storage of enhanced dark cutting beef in nitrite embedded packaging, increasing metmyoglobin formation upon repackaging. I hope I didn't butcher that too much.
Share the screen until he stops sharing the screen.
Oh, I stopped sharing the screen. Okay.
Sorry.
Sorry.
Okay.
Thank you. Sorry.
You're good. All right. Thank you, Dr. Mariana, for that introduction. Again, my name is Morgan Denzer, and I'm a PhD student in the Department of Animal and Food Sciences underneath Dr. Ramanathan. And today I will be presenting on the dark storage of enhanced dark cutting beef in nitrite embedded packaging, or NEP, increased metmyoglobin formation upon repackaging. So typically consumers want to see a bright cherry pigment in their steaks in a retail case because they associate this with freshness. And so when dark cutting steaks are seen, this can be negatively perceived by consumers, and therefore they're typically not used in a retail setting. They're more used for food service. And because of this loss of value, they're often discounted as carcasses. So in the United States, for example, approximately 1.9% of our carcasses are considered dark cutting, and the total cost of to our beef industry is anywhere between $91 and $251 million. So this is only in the United States. So if you think about worldwide and dark cutting beef, it can have a fairly significant impact on the beef industry and the profit for our beef producers. Therefore, dark cutting beef has been heavily researched in order to determine ways to introduce those products into a retail store setting. So previous research has focused on acidification as well as packaging. And so when you look at acidification, they use lactic acid to improve fresh color. However, this also increased what we call surface discoloration, or that formation of metmyoglobin, which is a brown pigment, which can be negatively perceived by consumers. Additionally, gluconadelta-lactone, or GDL, has been used to increase lightness of those dark cutting loins. Additionally, for packaging, matte packaging has been used in CO as well as HIOX systems to increase redness, and in combination with red rosemary, this was further improved. New technology called nitrite embedded packaging, or NEP, has started to be researched and utilized for dark cutting beef. And that's because it's shown to increase redness as well as color stability of these dark cutting steaks. Additionally, in combination with rosemary, there's been a further improvement of that red appearance. However, overall, it's had limited impacts on the dark appearance of these steaks. So although they're appearing red, they're still a dark red appearance. Therefore, nitrite embedded packaging in combination with acidification could have the potential to improve that overall color of those steaks. However, nitrite embedded packaging isn't anaerobic packaging. So with that, consumers typically prefer to see what we call PVC overwrap, or an aerobic system in the retail case. Therefore, this may not be the packaging that they would like to see in retail. Therefore, for our study, we hypothesized that a longer dark storage period in this NEP packaging with an acid enhancement will improve the retail color of enhanced dark cutting steaks upon repackaging them into a PVC system, or that preferred system for consumers. So for our objectives, we wanted to look at the various dark storage times and their impact on the color stability after those steaks had been removed from that NEP system. Additionally, we wanted to look at the effects of repackaging out of that NEP system on that overall retail color into PVC. So for processing, we use 6 normal pH loins and 8 dark cutting loins. For the dark cutting loins, they were bisected and randomly assigned to be non-enhanced or enhanced. And the enhanced loins had 0.5% gluconadelta-lactone and 0.1% rosemary as their final concentration. From there, steaks were sliced from all loins and randomly assigned to either 3, 6, or 9 days in dark storage. After dark storage and during retail display, instrumental color was evaluated using a Hunter Lab spectrophotometer. So for dark storage packaging, we placed those enhanced steaks into that nitrite embedded system, while the non-enhanced and the normal pH steaks were put into vacuum. So for retail display, again, the steaks that were selected for dark storage for 3 days, for example, were removed on day 3 and placed into PVC and held in PVC for 6 days for a simulated retail display. So this is followed for all steaks that were selected for days 6 and 9 in a similar procedure. For color evaluation, we looked at metmyoglobin formation, so again, that brown pigment, as well as delta E. And so delta E evaluated a change in color from hour 0 to hour 12 of retail display. For statistical analysis, the least square means were determined using the mixed procedure of SAS, and they were considered significant at a p-value less than 0.05. So first I want to take a look at the metmyoglobin formation for results. And we're talking about metmyoglobin, again, this is that brown pigment that can form on the surface that can have negative perceptions by the consumers. And so with this, a higher number will indicate more metmyoglobin formation. So looking from hour 0 to hour 12, those enhanced dark cutting steaks significantly increased in metmyoglobin formation, no matter the amount of dark storage time, indicating the limited effect of that dark storage time in the NEP system. Additionally, the enhanced dark cutting steaks had significantly more metmyoglobin formation than their normal pH in the non-enhanced dark cutting steaks. And then by the end of retail display, those enhanced dark cutting steaks had a very similar amount of metmyoglobin formation for those steaks that were held in 6 and 9 days of dark storage for the normal pH steaks. However, the normal pH steaks did have less metmyoglobin formation than the enhanced in comparison for those held in 3 days of dark storage. So again, overall, the dark storage time had limited effect on that metmyoglobin formation of those enhanced dark cutting steaks. So looking at the delta E evaluation, again, this is from hour 0 to hour 12, so just the first 12 hours of retail display. So with the normal pH steaks, they had a positive delta E. So this means that they increased in lightness as well as redness within the first 12 hours, which is expected because they're going through oxygenation or bloom to form that oxymyoglobin pigment. Additionally, the non-enhanced dark cutting steaks had an increase in delta E. However, that was numerically lower than the normal pH steaks, which is to be expected since they're going to be darker and less red than those steaks. For the enhanced dark cutting steaks, they had a negative delta E. So overall, this indicates that they actually became darker and less red within 12 hours of display in comparison to their initial color coming out of that NEP system. So in conclusion, repackaging out of the NEP system into PVC declined the red appearance as well as increased discoloration. Therefore, repackaging those enhanced dark cutting steaks at this time is probably not an option to improve color. However, we do know that NEP system can improve color of those dark cutting steaks. However, that air exposure was shown to reduce color stability. Further research then should evaluate the extension of color stability after repackaging from the NEP system. That's it. And at this time, and I will take any questions.
2 minutes for questions.
Morgan, so this is Danny Belmer. It's my understanding that dark cutting steaks don't represent any negative health impacts. So my question is, why spend millions of dollars and lots of research to make them look better rather than just educate consumers that dark steaks are perfectly fine? It seems like an education issue, but we spend a lot of effort fixing a problem that maybe isn't a problem.
Yes, you are correct. They do not have any health concerns. However, they do have some off flavors to them that can be unappealing to consumers. So while there is that appearance attribute that comes with education that might allow for consumers to select that steak, there are some of those off flavors that could then negatively be perceived by consumers that also would need to be evaluated to allow them to be truly marketable to all consumers.
Great, thank you.
Mhm.
Thirty seconds.
I'll just ask one more question, I guess. So if your packaging film, the nitride embedded packaging film was able to encompass all the product and without, it still has a problem with air, right, in the package?
Yeah, so it's considered anaerobic, just like the vacuum packaging, and consumers prefer to see that PVC overwrap packaging, so that styrofoam tray, and they're not as likely to purchase something that is in the vacuum package system. So since it appears like a vacuum package system, they wouldn't be as likely to select that product since they prefer the PVC.
Time.
Well, thank you very much.
Thank you.
All right, and our next speaker, they're really coming right on time there. I like that. Thank you. Carrie Meeks will be presenting on optimization of atmospheric cold plasma device for food decontamination. It's all yours, Carrie.
Thank you for the introduction, Dr. Mariana. Let me get this. Sorry, 1 second. Okay, so my name is Carrie Meeks, and I study underneath Dr. Maria Ma as a master's student. And today I will be presenting on cold plasma. Oh, hold on. Sorry, it's not wanting. Sorry. There we go. So there is many current decontamination methods, and they all have a pro to them, but they all also have a limitation. So for chlorinated water and microwave, they both affect the texture and the color, and that becomes an issue when we're processing fresh produce and fruits. In radiation, it works very, very well. I really like it, but it lacks that consumer appeal, which really limits us using it. And infrared heats the food quickly, which again would not be ideal for produce. And chlorine dioxide produces small amounts of toxic byproducts, which can be an issue for workers. And the electrolyzed oxidizing water is just a very high input method. It needs water, it needs continuous electrolysis and other additives. And organic acids is a good method, but it's very dependent on the concentration of the acid applied to the food, the pH of the environment, and the acid use. So it really is a true science. Dense phase CO2 is also a good method, but it's an expensive equipment in a large temperature range, which can make it difficult for industry to start up and start using that system. And the pulse light system only does clear packages. And as we all know, not every food is in a clear package at the store. And then ozone is limited because it does not react with water. So I think atmospheric cold plasma can fill in a niche here. It uses electricity, atmospheric air, and an accelerator to produce plasma. So there is no water and no chemicals added to this method. It has strong oxidizing properties, and its killing mechanism is by oxidation. It's dry and non-thermal, which is great for keeping food quality high. And it's low cost to set up, which will allow industry to adapt to this if they choose so to start using this treatment. However, it is highly variable on the efficiency depending on these following variables: distance, time, power available to the machine, and then the electrode width and the gap width between the electrodes on the accelerator and the generating pulse intervals. So the objective of this study was to assess the impact of electrode arrangements on the inactivation of foodborne pathogens by atmospheric cold plasma, and to investigate the influence of power input and pulsing intervals on the inactivation of foodborne pathogens by atmospheric cold plasma. So here on my methods on the left side, you can see what is like a cross-section diagram of the atmospheric cold plasma machine that we use. And up top here is our accurator, and you can see the electrodes and the plasma coming out. And then on the bottom, we have a Petri dish with a glass cover slip, and the lid is off so the plasma can reach the cover slip. So the methods that we did to perform this study was I took sterile glass cover slips and I spot inoculated them with a 5-strain mixture of Salmonella enterica, and that was about a 10 to the 9 colony forming units when I inoculated them. Then I air dried them for 1 hour before treatment, and then I treated them at a 2 centimeter distance for 2 and 5 minutes and a 5 centimeter distance for 2 and 5 minutes. Following the treatment, I immediately washed them in sterile 0.1% peptone water, and I plated the dilutions. The next morning, I performed an enumeration and a log reduction calculations, and the variables specific to this study was electrode width, gap between electrodes, treatment time, and distance of the item from the accurator. So my results, I took the cell counts from the enumeration plates and converted them into log values and log reductions. We found that the 2 centimeters for 5 minutes was the best overall for log reduction using a Wilcoxon sign rank test. The graph on the left is set at a distance of 2 centimeters, and each accurator is a little bit different in design, so that is why it's grouped out by accurator. But you can still see that every accurator still had a higher log reduction at the 5 minutes, even if it's not significantly, there's still a difference that you can see in the bars. And then on the right side, that graph is set at a time of 2 minutes. And you can really truly see here that a 2 centimeter distance really had a much higher log reduction compared to a 5 centimeter distance. In addition to just looking at the treatment, I also looked at the accurator C, which were performing better. And I got this data from the 2 centimeter 2 minute trial using ANOVA. The graph on the left has A1, A3, and A5, and they all have the same electrode gap of 0.5 millimeters. And we found that A3 and A5 was significantly better at log reduction compared to A1. And so we concluded on that a electrode width of 0.5 centimeters and higher has a higher log reduction. The graph on the right, the A2, A3, and A4 all have the same electrode width, which is 0.5 centimeters. But we did not find any significant differences between the electrode gaps in that one. So in summary, in this study, we found that a longer treatment time and a shorter treatment distance resulted in a higher log reduction among all of the accurators, and the electrode width at and above 0.5 centimeters improves inactivation efficiency significantly. But however, we did not find any significant difference in the inactivation efficiency among electrode gaps at 0.1, 0.5, and 1 millimeter. Next, we'll be looking into operational parameters such as power input and pulse intervals. While we were going through these trials, we have noticed that accurator A5 tends to use a lot more watts than some of the other accurators, so we'll be looking into that further also. Does anyone have any questions?
2.5 minutes for questions.
Hi, Carrie. This is Mark Vaughn. This is very interesting. I've heard about the ACP process for some time. Any plans to actually try it out on product? I know this is, hopefully that will be your next phase, is to try it out on product. And I'm in the produce business, so specifically that I would be interested there in that we have no kill step in produce, as you know, before most of it's consumed. So any plans along those lines?
Yes, so currently I am kind of just figuring out I'm using a cover slip to cut costs because, you know, produce is expensive. So I'm figuring out which has the highest kill, and then I will be using pecans to see how does it work on a rough texture type of produce.
Very good. Thank you.
Hi, Carrie. Pete Mariana here.
Mhm.
Just a question. I know, like, if it was UV, you have line of sight, so you're only going to be effective on the surface, the visible surface of a product. Does this kind of encompass a little bit more than just line of sight?
I believe it's about the same on that. It does just do the surface. So I think it is more better for produce and fresh fruits as compared to like meats and stuff like that.
Would it work on like a refrigerated product under refrigeration, or does it have a certain requirement of room temperature or something like that?
Far as I know, there isn't a requirement for room temperature. I haven't seen anything on that.
30 seconds for questions.
I guess the length of time that some of the process could be accommodated by a continuous flowing like conveyor belt.
Yes, that's kind of like our big end goal. We would like to have kind of a sealed in conveyor belt, you know, that can do it for a while. And then that way it'll be easy for those in industry to use this technology.
Time.
Okay. Well, thank you very much.
Thanks, Carrie.
Our next speaker is going to be Frank Kiemba. Is that close enough? Are you there? You have to unmute. Okay.
Yes, I'm here.
Okay, and Frank Kiemba will be presenting on differential aging-related changes in protein profiles of dark cutting and normal pH beef longissimus lumborum muscle. So it's all yours, Frank. Take it away.
You can all see my screen.
Uh-huh.
Yeah, thank you, Dr. Mariana, for that introduction. Again, my name is Frank Kiemba, and I'm a PhD student in the Department of Animal and Food Science here at Oklahoma State University. My presentation today is about differential aging-related changes in protein profiles of dark cutting and normal pH beef longissimus muscle. To start my talk, I would like to begin by giving you a little bit of introduction about dark cutting beef. Dark cutting beef is a meat quality defect in which meat does not display a characteristic bright red color we see in normal pH beef. For example, in this picture, you can see on the right we have a bright colored muscle, while on the left we have a dark colored muscle. And because of this color deviation, it is estimated that about $165 to $170 million is lost by the US beef industry because of dark cutting carcasses. In 2016, the National Beef Quality Audit estimated that about 0.74% of carcasses that were slaughtered were dark cutters. Therefore, this dark cutting beef has a wide occurrence, and in Canada, it is graded as a B4 quality grade with a 50% reduction in retail prices. The occurrence of this dark cutting beef is associated with defects in glycogen metabolism, and this is attributed to pre-slaughter stressful conditions. And as a result, we see that these dark cutting beef have a low capacity to accumulate lactic acid post-mortem, and therefore they have a greater than normal muscle pH, which is above 5.8. With this greater muscle pH, dark cutting beef can support mitochondrial respiration post-mortem, whereby the mitochondria can continue to consume oxygen while deoxygenating myoglobin, hence producing this dark cutting muscle. Previous research done in our lab has shown that actually, if you can age these dark cutters, you can improve the blooming characteristics of this dark cutting beef due to reduction in competition for oxygen with the mitochondria and myoglobin. And also, it's suggested that extended aging can, however, limit the capacity of meat to turn the brown form back to a bright red colored meat. However, the extent to which protein profiles in dark cutting beef compared to normal pH beef are regulated during aging is still unknown. Therefore, the hypothesis for this study was the changes in protein profiles during aging could modulate differences in characteristics between normal pH and dark-cutting beef. And our objective was to characterize aging-related changes in protein profiles in dark-cutting beef compared to normal pH beef longest mass muscle. Moving on to materials and methods, in this experiment, we utilized 6 normal pH and dark-cutting beef strip loins. These were collected from a local abattoir in Kansas City. And these were transported to the Food and Agricultural Product Center here at Oklahoma State University, where they were portioned into 3 different sections and aged at different ages. At each particular aging period, about 0.5 grams of muscle sample were collected from the normal pH and dark-cutting beef and were used for proteomic analysis as shown in this illustration. For protein extraction, we utilized the guanidine lysis buffer at a pH of 8.2, and protein concentration was determined using a tryptophan fluorescence standard assay. We used trypsin for protein digestion, and peptides were mapped using a liquid chromatography mass spectrometry based method. The obtained raw data files from the mass spectrometer were used to search the bovine proteome database, which contained 23,968 protein to identify the peptides. And we utilized full trypsin specificity with not more than 2 miscleavages, and the precursor iron mass of 20 parts per million with a tolerance of 0.3 Dalton was used for peptide identification. Peptides which had a mass probability score of a p-value less than 0.05 were considered significant. After database searching for statistical analysis and bioinformatic analysis, the collected raw data files from MaxQuant were exported into another software, Perseus, and these we did a differential protein expression whereby differences in protein ratios were considered significant at a false discovery rate of less than 0.05. To further understand more about the potential protein interaction between these differential expressed protein, we utilize a network analysis using Cytoscape. And moving on to the results, we observed that in this study, aging of beef has a differential regulation on protein profiles in dark-cutting beef compared to normal pH beef. For example, in this figure, you can see that on the y-axis, we have the number of identified differential expressed proteins, where the blue color here represents the total number of differential expressed proteins. The green color represents proteins that were downregulated, where the red color shows proteins with upregulation at a fold change greater than 1.4. In this study, we observed that at aging day 7, there was a greater number of regulated proteins in dark-cutting beef compared to normal pH beef, while as aging period increased to day 14, most of the proteins became downregulated in dark-cutting beef compared to normal pH beef. When we looked at differences in protein expression in dark-cutting beef at aging day 14 compared to day 7, we observed that there was a greater downregulation of proteins in dark-cutting beef compared to normal pH beef. However, evaluation of changes in protein expression in normal pH beef at day 14 and day 7 did not yield significant differences. To further validate this data, we also looked at principal component analysis shown in this figure, whereby on the y-axis we have component 1, which explains about 46.2% of the variability in our samples. The yellow color and orange color represents dark-cutting beef samples at day 7 and day 14 of aging, while the blue color and the purple color represents normal pH beef at day 7 and day 14. From this data, you can see that there was a clear separation in samples, especially in the dark-cutting treatment, whereby at day 7 and day 14, we can see a clear separation between our samples, while day 7 and day 14 in the normal pH beef did not show any significant separation. Consistent with this data, looking at the Histo clustering analysis, we can see that there was distinctive clusters in normal pH beef and dark-cutting beef proteome, and we identified 3 distinctive clusters. And here, the green color represents proteins with downregulation, while the red color represents proteins with upregulation. To further understand more about the differences, the potential protein interaction between these differentially expressed protein, we looked at protein interaction using a string database network. And the results revealed that differential protein stability in dark-cutting beef compared to normal pH beef. For example, at day 7, as you can see here, the red color represents proteins which had the upregulation, while the green color shows proteins with a downregulation. And when we look at this network, we can see that most of the proteins
2 minutes
that are
remaining.
connected, sorry?
2 minutes remaining.
Yeah, that are connected kind of had a greater abundance of proteins involved in energy metabolism and stress-related complexes. Therefore, we can say that the greater abundance of stress-mediated and redox proteins could suggest that there's a greater protein stability in dark-cutting beef compared to normal pH beef at day 7. Well, by day 14, most of these proteins became downregulated, and we observed that there was a greater abundance of complex 1 proteins, which could suggest that there's a greater capacity to produce reactive oxygen species in dark-cutting beef compared to normal pH beef. In summary, we can say that this study shows that aging of meat has a differential regulation mechanism on protein profiles in dark-cutting beef compared to normal pH beef. And we've shown that by day 7 of aging, dark-cutting beef has a greater abundance of proteins involved in energy metabolism, stress-mediated responses, and redox systems. While by day 14 of aging, dark-cutting beef had a reduced abundance of proteins involved in stress-mediated responses, glycogen metabolism, but with a greater abundance of complex 1 protein. With this, I would like to welcome your question.
40 seconds for questions.
Mr.
Hi.
Chumba,
Sorry, go ahead.
you go ahead. I'll wait.
Go ahead, repeat your question.
Okay, Mr. Chumba.
Yes, sir.
As far as the protein expression, the downregulated gene proteins, are you going to explore them further to identify what specific genes are associated with those?
I beg your pardon, I didn't get the last part.
What genes are associated with activation or deactivation of those genes, the downregulation of those genes? What's causing that?
So, yeah, thank you for your question. Yes, we did identify these proteins that were connected to the downregulation, and we see that by day 14 of aging, we had a decrease in the HSP90 pathway protein and the protease system pathway.
All right, well, thank you, Frank. Any further questions, you can contact Frank directly. I'm sure he'll be happy to answer them. Our next presenter, Amandeep Kaur. Close, you got to unmute. Okay. The effect of low temperature treatments on Pistillate pecan flowers. It's all yours.
Thank you. Hello everyone, my name is Amandeep Kaur and I'm a master's student in Department of Horticulture. I'm working as a graduate research assistant under Dr. Lu Zhang. And my topic for today's presentation is the effect of low temperature treatments on Pistillate pecan flowers. So starting with pecans, pecans are deciduous tree and they are native to North America. USA is the #1 producer of pecan as it contributes about 80% of world's pecan market. And pecans are famous for their nutritional and medicinal value. As you can see in this picture, the blue ones are the major states of US that are producing pecans, as with New Mexico and Georgia are the top producer, and Oklahoma comes at 5th place in pecan production. And this is a picture from our pecan orchard in Perkins. So pecan trees have some special characteristics, such as they are monoecious plant. That means they have both male and female flower on the same tree, but they have at different places. So for example, the male flower is from the 1-year-old branch and they are many in number, while the female flowers produced from current year's top terminal buds, and they are few in number. And also pecans have dichogamy, two type of dichogamy. That means their male and female flower mature at different times, and because of that dichogamy they have the cross pollination. So during the spring, pecan buds start to grow and break their dormancy. And in April, we have, at the end of April, we have flowers. So during this stage, pecan buds are considered as the most susceptible stages to any environment changes. And the spring freeze during this time can cause a major loss to pecan production and to other crops also. For example, we have the Easter freeze in 2007, which occur in the first week of April. And during that time, most of the tree were at bud break and bloom stage. And that caused a very severe damage to a wide range of a wide region of Eastern US. So these are some pictures of pecan bud and inflorescence damage by the spring freeze. So this one is a picture from last year's spring freeze event. And in this table, we are representing the number of hours with temperature less than 4 degrees during the month of April and May. So as you can see, in 2008, we have spring freeze, and even 2020 also, we have 83 hours with temperature less than 4 degrees Celsius. And in the right picture, you can see damage to the male flowers after the freeze event. So objective of our study is to find the threshold temperature and tolerance range of pecan buds and flowers to spring freeze. And for this experiment, we are using 3 cultivars, Kenza, Pony, Miramic, and 2 growth stages, the outer bud scale shed growth and the bloom stage. And for our experiment, we are using 4 different temperature treatment from -6 to 4 degrees Celsius and 2 different duration. So we use the Conviron 8 freezing unit to give the temperature treatment, and after them we grow our branches in the growth chamber using the historically mimicked temperature and humidity conditions of spring, as you can see it in this table. So after bloom, we observe the structure of stigma. So this is how the female flowers of pecan looks like. So we study the stigma structure after the bloom by using scanning electron microscope. As you can see, this is the pecan flower, and you can see this here in scanning electron microscopy. So we use our microscopy laboratory for this experiment. So after we observe that at outer bud scale shed, the temperature treatment of -6 and -4, there was no leaves and female flower development. As you can see in this picture, even the 4 degrees Celsius for 4 hours, there was no leaf and female flower development. And at the bloom stage, the cold temperature treatments, even 4 degree for 4 Celsius, sorry, 4 degree Celsius for 4 hours, cause the collapsing of papillary cells of stigma. These cells are present at the stigma surface, and these cells are act as a platform for the adhesion of the pollen. So if there is a damage to these stigma surface cells, then the pollen will not be able to attach to the stigma surface, and there will be no successful pollination and fertilization. So at the summary, we observe that there is a poor development of buds and flowers by -6 and -4 degrees Celsius treatments. And also we observe that there is a wide range of temperature than our previous perception that can affect the pecan flowers. For example, we don't consider 4 degrees Celsius even for 4 hours as a freezing temperature, but we saw effective damage by this temperature. So in future, we are going to continue observe the stigma surface via different temperature treatment, and we also observe the stigma receptivity and ovule viability. So in the end, I would like to thank my major guide, Dr. Lu Zhang, and my committee members and my lab personals for helping me in this experiment, and also the funding by Oklahoma Department of Agriculture, Food and Forestry Specialty Crop Grant Program to provide funding to our project. And these are my references. Thank you. I would like to have your questions.
3 minutes for questions.
Okay, hi, Amandeep, this is Lu. Thank you very much for this great presentation. So my question is regarding the stigma receptivity. You mentioned that you use SEM to check the popular cell. Yeah. And in the next year, actually is this coming year, I know you have already collected some samples, and for the stigma receptivity, can you further discuss what type of methods you will try to use?
So for stigma receptivity, we are going to try 2 different methods. So first method involves the benzidine and hydrogen peroxide solution. So when we use this solution and we put that solution on fresh stigma, so it will create a color and also the bubble, and we will observe that under microscope. So the intensity of bubbles and color indicate whether the stigma is viable or means receptive or not. And second method is also almost same, but in that we vary the chemicals, like the test solution that compose of phosphate buffers, nitrosolium. So the rest of method is same. We will just change the chemicals.
Thank you.
Thank you.
Minute and a half for more questions.
Well, I mean, I have a question. I mean, well, not really a question, but I guess for pecan trees, they're so large, you're really hard pressed to do anything physical. I've seen a couple of movies lately where small fruit trees, I guess most of the fruit trees they have with orchards are pretty small, and they light little fires in there when they have a cold front coming in to kind of keep the temperature at the ground level up a little bit. You really can't do anything like that, right, with pecan trees?
Is it about the temperature difference?
Well,
yeah.
you're talking about the cold weather. If you have a cold front comes in, it ruins the bloom.
Yes, so we have mostly the radiation freeze in the pecan. That means it damage more at the bottom of the canopy than the upper part, because they are very big trees. So there is a difference in temperature at the bottom and the upper canopy. So it causes more damage to the bottom canopy as compared to upper one.
Oh, okay.
20 seconds. We can move on if you'd like.
All right. Well, thank you very much.
Thank you.
We'll go to our next speaker, Nicholas Lopez. Nicholas?
Oh, can you hear me, Dr. Mariana?
Can't see you, but I can hear you.
Yes, I just. Okay, let me. I don't know what's going on with my webcam, but okay.
There you are. Okay. All right. So our next speaker is Nicholas Lopez, evaluation of whole genome sequencing for rapid identification and molecular characterization of E. coli, Escherichia coli isolates from food and the environment. So it's all yours.
Thank you, Dr. Mariana, for your introduction. Let me share my screen now. Okay. Okay. Can you see my slides now?
Yes.
Yes. Okay. So good morning, everyone. My name is Nicholas Lopez. I am a food science master's student under the tutelage of Dr. Lima. So I will present part of my research project. So first, why do we care about E. coli? So E. coli is commonly a commensal bacteria found in digestive tract of different animals. However, due to the selective pressure of certain niches, some strains have adopted pathogenic properties. So how can we identify these pathogenic strains? We can characterize their O and H antigens. Also, we can identify the presence of different virulence factors as well as antibiotic resistant genes. And we can also study the relationship between different pathogenic strains based on phylogenetic analysis. So welcome everybody to the whole genome sequencing era. As you can see here in this graphic, due to the advances in technology, it's becoming cheaper and cheaper. And that's the reason why the CDC and the FDA have adopted it as a gold standard for foodborne bacterial pathogen surveillance in their surveillance networks. And what's the advantage of using this technology? So here we have the whole picture of the bacterial chromosome, which allows us to identify the presence of different genes and other mechanisms of regulation. So the mostly used platforms for whole genome sequencing are Illumina, Oxford Nanopore technology, and PacBio. Even though Illumina has the highest accuracy of these technologies, ONT and PacBio can offer longer reads. In addition to that, Oxford Nanopore technology can provide us portability, and also we can pull out the reads on real time. With that being said, the objective of my study was to evaluate the performance of mini-ion sequencing for rapid identification and molecular characterization of E. coli isolated from food and environment. So for doing that, we collected 11 isolates from Pecan orchards following the bacteriological analytical manual. We screened the presence of 3 different virulence genes, and for our isolates, we performed the whole genome sequencing using the Oxford Nanopore technology mini-ion and the NextSeq Illumina platforms. After that, we perform a quality check of our reads. And in addition to that, we used a custom Perl script in order to subsample the mini-ions reads based on when they were obtained. With those reads, we perform a species confirmation. We also perform a serotype prediction. And we could also identify the determinants of the genes Shiga toxin variant 1, the Shiga toxin variant 2, and also the intimin gene. Finally, we analyzed the continuity of the genomes that we could assemble from those reads. So we assembled the reads separately from Illumina and mini-ion, but we also created hybrid assemblies. With the quality of those assemblies, we perform a distributional independent multivariate analysis. And we also characterize the virulence factors and antibiotic resistant genes in those genomes. So for doing that, we perform the annotation of those genomes. And with the annotated proteins, we perform a BLASTP search against the comprehensive antibiotic resistant database and the virulence factor database. So moving on with my results, as we can see here for the species confirmation, from 15 minutes, we could identify the expected species, that in this case was E. coli. For the serotype prediction, from 8 hours, we could identify the O antigen in 10 of the 11 isolates. However, for the H antigens, we could only identify it in 4 of the 11 isolates, even if we use the full reads data set. So if we could identify the presence of the virulence genes in 7 of the 11 isolates, from which after 24 hours, we could identify the determinants in 5 of these isolates. So finally, we analyzed the continuity of the genomes that we assembled with those reads. From 4 hours, we obtained significantly similar results to those obtained with the full reads data set. And for the characterization of the virulence factors and antibiotic resistant genes, likewise for the continuity, we obtained similar results from 4 hours in comparison with the full reads data set. However, in comparison with the results obtained with the hybrid assemblies, we obtained significant differences in those hits that were obtained from the BLASTP search. So in summary, comparing to Illumina, whole genome sequencing by ONT mini-ion offers the potential of real-time identification and characterization of potential foodborne bacterial pathogens. So the species level identification that we achieved here was at 15 minutes of the sequencing run. The O antigen prediction was achieved after 8 hours. the variants of the virulent genes were detected after 24 hours, but Oxford Nanopore technology mini-ions loss precision limits its ability to characterize fully factors that might be relevant for the E. coli assessment. Thank you so much. I would be really happy to hear any opinion, suggestion, or question that you may have.
4.5 minutes for questions. We have 4 minutes. Questions, anyone?
I'll say, Nicholas, why does it take so much longer for the other identifications than it, I forgot which ones you had there that took so many hours. Whereas I know species, I guess, would be a little bit easier, but some of the antigen identifications that took hours to achieve, what is the reason for that?
Okay, let me think. As far as I know, I think that it's because of the error rate that mini-ion has. So you need certain coverage to cover to at least cover specific genes in comparison to the genes that are for the O antigen or for the H antigen identification. Because as you may know, for the O antigen, there are more than one genes involved for the production of the lipopolysaccharide and also for the transportation of it via transmembrane proteins. So that's the reason, because there you are identifying more than only one gene. So in that way, you can predict the O antigen easier than in comparison to identify only one virulence factor. In this case, one determinant for the Shiga toxin variant 1 or for the Shiga toxin variant 2.
Okay.
2.5 minutes for more questions.
I used Fernley was the oxygen, the portable sequencing instrument for this study.
Oh, the name is mini-ion. Mini-ion is a device produced by Oxford Nanopore, and it's pretty simple to use because you can only plug it into a USB port in any computer. So you don't even need like an electrical source. You can even use it if you are in the rainforest or if you are in the mountain or even in the Arctic. It's like the new, it's the third generation of sequencing. It has like these advantages because it's feasible to be used anywhere, almost anywhere. So
Thank you.
Was that your question?
Yes, thank you.
Okay.
1.5 minutes, one more question.
Hi, this is Nurhan Danford. I'm wondering about the practical implications of time at the molecular characterization level. So we're looking at minutes versus hours. So do we really need this kind of information in minutes, or is that cost-related issue?
Oh, okay. So when you are characterizing a bacteria, it's because it has been already detected in a foodborne disease outbreak. So the time matters there, because if you do it like quickly enough, you can save a lot of lives. So the turnaround time, if you reduce it, it's better. It's better because you can reduce also the time that you send the sign for the outbreak, and you can take better measures to prevent the spread of it.
Thank you.
You're welcome.
30 seconds. Let's move on.
Okay. Well, thank you, Nicholas.
Thank you, Dr. Moreno.
Our next speaker is Elena Gerhart, and she will be presenting on reduced lignin alfalfa in water-limited environments. It's all yours, Elena.
Thank you. I'm not able to share my screen.
It's because I am sharing it. Okay.
Okay, perfect.
Sorry.
No, you're good. Well, thank you for that brief introduction. Like you said, my name is Elena Gerhart, and I am with the Plant and Soil Science Department here at Oklahoma State University as a master's student. And today I'm going to be talking to you about reduced lignin alfalfa in water-limited environments. So alfalfa is known as the queen of forages because it provides several attractive qualities. One is that it's a high-yielding forage and it produces a high nutritive value, and is found to be very palatable, which makes it an asset to both beef and dairy industries. However, despite being the queen of forages, we have seen an overall decline in production area in the last 10 years, both nationwide and in Oklahoma. As you can see in the graphs on the right-hand side, the US alfalfa production area has declined by 16%. Now, looking in the Oklahoma alfalfa area production, we've seen it doubly reduced, as we have seen a reduction of 33.8% since 2010. Now, this could be due to a couple of different factors. Alfalfa might have to compete with other crops, or other industries that rely on alfalfa might be in an economic downturn, making it not as viable to use as a feed source. But one thing that we found in Oklahoma is that there's just not a lot of information on the new alfalfa technologies to producers. So they're not able to make best management practice decisions that include new technology that could increase the area of alfalfa produced. However, in other locations, they have found that reduced lignin alfalfa, which is a genetically modified variety to accumulate less alfalfa or less lignin in the alfalfa, can increase harvest intervals, and it can also maintain similar quality and quantity to that of conventional varieties. This led us to developing this study in Oklahoma, which is a water-limited environment, and has not, reduced lignin alfalfa has not yet been studied in this setting. So we wanted to evaluate the forage quality and quantity of a reduced lignin alfalfa variety, and then compare it to conventional varieties that are already being produced here in Oklahoma. So for this experimental design, we decided to truly evaluate it would be the best, to do that best would be to select 3 different harvest intervals that are commonly found across the state. In this picture, you can see the 3 harvest intervals that we selected were 28, 35, and 42 days, and we use those as our main plots. Within each main plot, we then selected 4 cultivars. The first one, which is HVX, is our reduced lignin alfalfa cultivar, while also containing the Roundup Ready gene. In order to have a fair comparison, we also selected 3 non-reduced lignin alfalfa varieties that contain the Roundup Ready gene. This allows them to be most similar and allow us to not have a lot of variability in our comparison. All cultivars were selected on the basis that they are commonly found in Oklahoma already and are all genetically similar. So moving on to our results, as I mentioned, alfalfa is known for its high yielding capabilities. Now, over time, alfalfa is known to increase. On your left-hand side, you're going to see our harvest interval data. And typically, as harvest intervals increase, you're also going to see an increase in biomass production, or above ground dry matter. And we found these results to be similar with other studies conducted in non-limited water environments. However, we did see an interesting fact that HVX, our reduced lignin alfalfa cultivar, did have a slight reduction and was statistically different than two cultivars in Lahoma, one of our locations. However, we only saw a statistical difference between 1 cultivar in Stillwater. Now, this could be due to the fact that soil conditions are slightly different in both locations, and one is more suited for alfalfa production over the other. Next, we're going to talk about one of the quality factors that producers look for when selecting an alfalfa cultivar. ADL is a measurement of lignin, and lignin is needed for structural integrity in the plant. And it typically increases as regrowth occurs. The plant is maturing and needing to hold the stem up, and lignin accumulates for that reason. So typically we will see an increase of lignin as days of regrowth occur. And we found that our results are similar to previous studies in the upper Midwest. Because we're looking at reduced lignin alfalfa cultivars, we would assume that we're going to see a reduction in lignin for the HVX variety, which is our reduced lignin variety. And we did see a statistical difference between the HVX and all other cultivars in one location, Lahoma. However, we only saw a slight numerical decline when looking at the Stillwater results. We were kind of perplexed by that, and we went back and looked through literature to see if other people had found an explanation or reason for that. And we did find that in drought-related events or high soil moisture stress, that there is going to be a reduction of ADL concentrations in conventional alfalfa cultivars, which might be why we don't see such a big difference like we did in Lahoma. Next, we're going to talk about crude protein. Crude protein is known to decline as days of regrowth occurs. So our results in both Lahoma and Stillwater for our harvest intervals are consistent with that of other studies. Now, when looking at the cultivar selection, we do see that HVX in Lahoma was statistically higher than the other cultivars. This is due to the dilution effect, which means that by removing some of the lignin, we are going to be able to put more protein into the HVX cultivar. But because we did not see as big of a reduction in the lignin concentrations in Stillwater, I believe that is why we did not see such, we only saw a numerical difference in Stillwater between HVX and the other three cultivars. Lastly, the last quality factor that we're going to be talking about today is in vitro dry matter digestibility after 48 hours. This is a measurement of how much of the crop that we are feeding to ruminants is going to be used within 48 hours. How much is going to be digested? IVTDM, or in vitro dry matter digestibility, is closely related to the amount of lignin in the plant. If you remember, lignin increases as days of regrowth occurs. Similarly, IVTDM is going to be seen to decline because of the close relationship. We found that our results of IVTDM were similar to other studies that did find a decline in digestibility over days of regrowth. However, we would assume that because the HVX variety did have a decrease in lignin, we are going to see an increase in digestibility. We did see a statistical difference between the HVX variety and all other cultivars in Lahoma. However, once again, coming back to the ADL concentrations, we didn't see a large difference there, which is why I believe we did not see a statistical difference in Harv Extra between the other three cultivars in Stillwater. Now, one thing to keep in mind is that this is a first-year alfalfa study, and alfalfa, as it is a perennial crop, will continue to be in production for following years. Lignin tends to increase as the age of stand increases, so I do think that in future years of this study, we will see a little bit of a difference. I want to leave you with a few conclusions that we found that 35 and 42-day harvest intervals were not statistically different, but we did result in a higher forage quality for the 35 days. We did see a reduction in production for the 28-day harvest interval, but we did see a much higher forage quality. Lastly, coming back to the reduced lignin alfalfa cultivar, we did find it to have less production and yield, but we had a higher crude protein, higher IVTDM, and a lower ADL. The next steps of this study will be to conduct an economic analysis and calculate water use efficiency. At this time, I will take any questions.
1 minute for questions.
Hi, Elena. I'm Carrie, and I have a question for you. So, is this lignin just based off of for digestibility, or is it also for alfalfa producers? My family grows some, and I know after a couple cuttings, it gets really woody. Would that also be a pro to having this variety?
Yeah, so what you're seeing, the woody concentration, that's definitely going to be your lignin. And as the age of the stand gets older, you're going to see more of that. Okay, so using a reduced lignin alfalfa would help with that.
Okay, thank you.
Yeah.
30 more seconds.
This is Nurhan Danford. As the lignin amount decreases in the plant, I saw that some of the original or conventional varieties, there was not much difference in the protein content. So what is that decrease due to? I guess there's a positive correlation with the protein content, but if there's no difference in protein, is that the carbohydrates increasing? Or?
Yeah, so when we see more lignin or other things, we're not going to see as big of a difference in crude protein. However, I would say that crude protein doesn't tend to vary a whole lot within alfalfa. We're typically looking more for like fibers or lignin, so that way we can increase digestibility. I wouldn't say that crude protein tends to vary too much.
And that's time.
Thank you.
Okay. And thank you very much, Elena. And our next speaker is Caitlin Karolinko, speaking on the use of alternative salts in Biltong marinade to reduce sodium and still achieve USDA FSIS 5-log reduction of salmonella. It's all yours, Caitlin.
Perfect, thank you. Okay, so I am Caitlin Karolinko. I am a PhD student in the Department of Food and Animal Science. And like Dr. Mariana said, I am going to be talking about alternative salts in the Biltong marinade to reduce sodium, as well as to achieve that 5-log reduction as suggested by the USDA. So what is Biltong? It is a South African style beef jerky. It usually uses lean meat that's marinated in traditional spices like coriander, as well as vinegar and salt, and then basically dried at room temperature and humidity. Biltong does not use high heat and high humidity as required by USDA beef jerky guidelines. Therefore, producers of Biltong still need to demonstrate that their product is a microbial safe product for consumers. They have 2 different ways that they can demonstrate this. One would be to demonstrate a 2-log reduction of salmonella. However, then they're also required to test every ingredient that goes into that process. It's both time consuming and costly for manufacturers. So a more beneficial option would be to just demonstrate a 5-log reduction without any additional ingredient testing. So ready-to-eat meats like Biltong commonly use large amounts of salt, not only to flavor the product, but also to help limit the microbial growth. However, consuming large amounts of salt in your diet can lead to negative health effects, including high blood pressure, heart disease, obesity. Previous work in our lab has demonstrated that we've been able to achieve that 5-log reduction using sodium chloride within the marinade. However, the USDA does not allow for equal salt replacement. So you can't substitute that salt with a different salt unless you can prove that that new salt is just as effective as the originally invalidated ingredient. Therefore, the goal of this project was to try to use alternative salts such as potassium chloride and calcium chloride in the Biltong marinade to achieve that 5-log reduction of salmonella. So in order to make the Biltong, we inoculated beef pieces with a 5-Saravar salmonella cocktail. These beef pieces were then vacuum tumbled in a marinade consisting of spices, vinegar, and then one of the 3 salts, and then hung to dry for 8 to 10 days, basically room temperature and humidity. We sampled the beef pieces every 2 days of drying to enumerate the remaining salmonella left on the beef pieces. And this is the results from that. This dotted line down here is the target 5-log reduction from the initial inoculum level. All 3 salts were able to achieve that 5-log reduction. Calcium chloride was able to achieve it by day 6. Sodium chloride was able to achieve it by day 7, and potassium chloride was able to achieve it by day 8. The process using sodium chloride and calcium chloride were not significantly different from each other. However, both processes were significantly different from the process using potassium chloride. We also wanted to quantify the sodium, calcium, and potassium ions found in our Biltong. We used these ion-specific electrometers in order to do that. We first wanted to validate how accurate these meters were, so we did standard curves for each one of them. You can see from the high R-square value, it's almost close to 1, indicating that these meters are actually very accurate. So after the Biltong was done drying, we homogenized it in a laboratory blender, macerated it with distilled water, and then that liquid sample was what was used in these meters to quantify the ions. And this is the results from that. Basically, what we found was that the ion corresponding to this ion salt was the ion that was the most abundant. So in other words, the Biltong that was made with sodium chloride, the sodium ion was most abundant in that sample. We also found that potassium ions were elevated across the board regardless of the salt used. So we wanted to kind of determine what that source of the potassium ion was. Therefore, we did a run of biltong with just beef, no additional salt, spice, or vinegar to see if we could isolate it to or correlate it to the beef. And then a biltong run with just spice and vinegar, no salt, to see if we could correlate it to the spice and vinegar. And what we found was the elevated levels coming from just the beef alone biltong was the source of the potassium ion itself. We also wanted to look at how our biltong compared to commercially available biltong. So we looked at 2 different companies that produce it, as well as 2 different flavor profiles within each company. We found similar levels of elevated potassium levels, presumably coming from the beef again itself. We found that both commercially available and our biltong had low levels of calcium. And then our levels of salt were similar in both the commercially available and our in-lab made biltong. We did notice using those ion meters, though, that some differences in the reported or the stated levels of sodium on the packaging on the nutrition label. We had found that actually company B had overstated the amount of salt. So basically what was listed as the sodium in the salt on the nutrition label was actually higher than what we found with the meters, and the opposite with company B. So it was actually understated. So they were reporting less salt. We actually found more salt in their product than they were actually reporting. So final conclusions from this, we are able to achieve that USDA recommended 5-log reduction of salmonella using either potassium chloride or calcium chloride in the biltong marinade. This will allow manufacturers to market biltong as a healthier, lower sodium option while still producing a microbial safe product. In the future, we want to look at the sensory or do a sensory analysis of the biltong. Some literature has shown that calcium chloride and potassium chloride, when used at high concentrations, can actually produce like a metallic off taste to food products. So we kind of want to do, while yes, we're producing a microbially safe product, we actually want to make sure it tastes good to consumers. So by doing a sensory panel, would see if the marinade flavors are covering that, or if that metallic present is present in our samples. The significance of this being that now biltong producers have more options with salt. They can now use sodium chloride as well as potassium chloride and calcium chloride within their biltong product to produce a microbial safe product for consumers. So I'll take any questions.
4 minutes for questions.
Hi, this is Nurhan Danford. Sure. I guess that the whole purpose of this process is to reduce the sodium amount, right? Because of health concerns. So I was wondering, how would you rate the potassium versus calcium in diet in terms of health effects?
So both are lacking usually in diets today. So this would kind of be actually, by using these alternative salts, would give it a boost in your diet. I don't know that one's better than the other, if that's kind of what you're more asking. Potassium versus calcium, I think they both have equal health benefits. It just allows it to not have a more heart healthy option than using the sodium chloride.
So are you saying that they don't have similar effect on blood pressure?
The calcium and the sodium, or the calcium and the potassium?
Okay, yeah.
Yeah, no, they don't have the same effect on the heart as the sodium does.
Okay, thank you.
Yep.
2.5 minutes for questions.
Hi, Caitlin. This is Morgan from the Animal and Food Science Department. I just had a question. I've tried biltong before, and it has a very vinegar base. And so with that different characteristic in comparison to our American jerky products, how do you think that sensory profile would possibly play out to consumers beyond possibly the traditional African market?
Sure. So I think it definitely has a different flavor profile. I mean, I've had it. I kind of like it more than American beef jerky. It does have a more acidic taste to it. Also, it's more chewier than beef jerky. I think it'd play pretty well. I think consumers have a pretty open mind when it comes to beef. So I think it would play out pretty well.
1.5 minutes for questions.
I'll just add to, the comment to Morgan, or reply to Morgan, all the processors that we've worked with in regards to assisting them and their validations. I mean, it's not that we're supposed to release a private company business, but they've all expanded their operations in their Biltong operations, adding on, getting additional facilities, increasing facilities, and it sounds like they're all doing fantastic with this particular product line in the US. So maybe it's not even close to being saturated yet. So
let's move on.
All right. Thank you. Our next presenter, thank you, Caitlin. Our next presenter is Kavya Gavai, and she'll be presenting on microbial validation of Biltong processing to achieve 5-log reduction of Listeria monocytogenes and E. coli 0157H7. So, Kavya?
Can you hear me, Dr. Mariana? Hello?
I can hear you, Kavya.
Okay. Hi, good morning, everybody. I'm a graduate student, graduate master's student working in the Department of Animal and Food Science. So today I'm going to present on the microbial validation of Biltong processing to achieve 5-log reduction of Listeria monocytogenes and E. coli 0157H7. As we have already talked about Biltong, we know that Biltong is a ready-to-eat dried meat product from South Africa. It is similar to beef jerky, wherein we use the lean beef strips that is marinated in salt, spice, and vinegar and dried at an ambient humidity and temperature. So it has become one of the most popular choices for the consumers because it is a favorable go-to snack food, and it is rich in protein, which has increased interest to manufacture the Biltong here in the United States. However, it requires USDA FSI's validation to be manufactured here in the United States. So addition of the salt mainly decreases the water activity, increasing the shelf life of the product. And addition of the vinegar inhibits the microbial growth and influence to the flavor and the water binding capacity. So beef jerky is manufactured here in the United States under certain guidelines, including maintaining the relative humidity greater than 90% throughout cooking. And the product must be cooked at a temperature greater than 175 degrees Fahrenheit. Since Biltong is not cooked, it doesn't meet the above mentioned guidelines. So the processor must demonstrate to USDA that their process leads to sufficient reduction of the pathogen. So in order to do this, they have two options. The first one is that the process must demonstrate a 2-log reduction, and one must test every ingredient to make sure that there is no pathogen. And the second one is that the whole process must demonstrate a greater than 5-log reduction of the pathogen. So in our lab, Dr. Mariana always wants us to make sure that we are using the right strains before starting our experiment. So we examined Listeria monocytogenes on Mox agar, which gave us black colored colonies with a concave shape. On the other hand, we also examined E. coli 0157H7 on Chrome agar 0157, which gave us the mauve colored colonies. Both the strains of Listeria monocytogenes and E. coli 0157H7 are resistant to antibiotics. Listeria monocytogenes is resistant to streptomycin and rifamycin, whereas E. coli 0157H7 is resistant to rifamycin and novomycin. Talking about the processing of Biltong in order to achieve 5-log reduction, we initially grew our bacterial strains that are acid adapted. Acid adaptation mainly makes these strains less susceptible to the injuries during acidic conditions like addition of vinegar in the marinade. So these bacterial strains were inoculated onto the beef pieces, and rubbed using the gloved finger technique, followed by dipping in water for 30 seconds that mimics the commercial dip treatment. Then the beef pieces were marinated for 30 minutes in a traditional marinade, including spice, salt, and vinegar under vacuum pressure, and dried in a temperature-controlled humidity oven for 10 days. So now that our beef is vacuum tumbled, it is a non-intact beef. Other example of non-intact beef are a hamburger and blade tenderized beef. The biggest concern with the non-intact beef is that during the process, the bacteria gets translocated internally into the meat. So in order to combat this, the product is cooked to ensure the safety. So as we know that biltong is not cooked, the safety mainly depends on maintaining the low water activity. USDA recommends that the water activity for the biltong should be less than 0.85 in order to prevent the enterotoxin formation should Staphylococcus aureus be present. So we can see that during processing of biltong in our experiments, the water activity maintained after 10 days was 0.79 for Listeria monocytogenes and 0.8 for E. coli 015787, that is less than 0.85 as recommended by USDA. So here we have the results for the overall reduction of Listeria monocytogenes for the combined components of the marinade, and we were able to achieve the targeted 5-log reduction after a drying period of 6 days, and greater than 6-log reduction after a drying period of 10 days. We also wanted to look at individual components of the marinade so that we also wanted to look at the individual components of the marinade, wherein we have that vinegar alone was able to achieve a 5-log reduction, which is similar to the reduction obtained from the combined components of the marinade. So coming to E. coli 015787, we have also achieved a targeted 5-log reduction after a drying period of 4 days. Coming to the individual components of the marinade, both salt and vinegar played an important role in overall reduction of the pathogen, and the similar reduction to that of the combined components of the marinade. So in conclusion, we were able to achieve greater than 5-log reduction and greater than 6-log reduction for E. coli 015787 and Listeria monocytogenes respectively with a subsequent drying period of 10 days. No one previously was able to achieve 5-log reduction besides Dr. Moriana's lab. This is mainly because of the method of enumeration followed. Others have followed CFU per gram as a method of enumeration. However, the meat loses 60% of its moisture at the end of the process. So using CFU per gram basis method of enumeration increases the residual counts, which goes against the log reduction. So in our lab, we followed the method of enumeration to be CFU per ml, wherein we used fixed volume diluent, which eliminates the issue regarding the loss of moisture of the beef to the microbial count. Thank you. Any questions?
3 minutes for questions.
How large is the market for biltong?
Sorry?
How large is the market for biltong as far as popular choices for consumers?
I think it is very huge when compared to the beef jerky, because we know that it is a ready-to-go snack food, so it is highly preferred. So it has a lot of preference.
All right, thank you.
2 more minutes for questions.
Hi, this is Nurhan Danver. The data analysis, your CFU per milligram versus milliliter versus gram. So which one is acceptable? Both are acceptable in terms of data analysis? Because the numbers are different, right? The results are
Yes.
different. Yeah.
Yes, I think CFU per ml is the right one because we can see that the residual counts are not increased because we used fixed diluent. We use 100 ml of NBPW in our experiment, which mainly eliminates the issue regarding the moisture loss and the microbial count. So I think CFU per ml will be great. And it doesn't go against the log reduction.
Thank you.
1 more minute for questions.
I'll just ask Kavya, what organism is next? So we validated Salmonella, Listeria, E. coli 0157, what else?
Yeah, we're going to validate for Staphylococcus aureus next. So
Okay.
pretty excited for that.
So USDA really only requires, for a validated process, only requires a pathogen of concern, which we accommodated with Salmonella. What is the need for all these other validations? Who will that help?
So because many other companies also want to look for these particular organisms, not just Salmonella. So they also want to look how, in terms of E. coli 015787, Listeria, Monocytogenes, and next, as we're working on Staphylococcus aureus. So the companies are very much eager to know the studies on these particular organisms also.
And that's time.
All right, well, thank you very much.
Yep.
Okay. Our next speaker is going to be Sriram Mikalineni.
You got it.
Mikalineni.
Yes.
Sorry about that.
It's fine.
Okay. Oh, I see a different name there. Okay. And he will be presenting on identification of quality parameters in manufacturing edible beef skin. Okay, we know all about that here in FAPSI.
Thank you, Dr. Mariana. I think you can smell it every Friday.
I didn't want to say that, but yeah.
So, good morning, everyone. My name is Sriram Mikalineni. I'm a graduate student under Dr. Tim Bowser in FAPC. Today, I'm going to present about a study of manufacturing edible beef skin, which we call it as Kanda K Pomo. I'll explain why we called it with that name in the next slides. Here in the first slide, you can see the raw hides, and this is the final product after vacuum packaging. There you go. So first, I would like to give some introduction about Pomo. So it's a very popular skin meat in Southwest Nigeria. So popularly it is called as Pomo, K Pomo, and Kanda in Southwest Nigeria, and Wile in West African countries like Ghana. So our client collaborated all the names to just for identification and named it as Kanda K Pomo. So Pomo is loved by all regardless of society classes just because of its unique taste and texture. There are 2 types of Pomos available in Nigerian market. One is white Pomo, another one is brown Pomo. So they're just defined by how they are processed. So white Pomo is processed by removing hair using razor blades or by hand plucking, and then followed by boiling in water for a couple of hours. And the brown Pomo is processed by singeing with fire by the main source of using tires, petrol, and kerosene and wood burning. So because of this burning tides, it may lead to contamination of the Pomo by having some dioxins and polyhydrocarbons on the final product. So Nigerian government decided to ban this Pomo, and then there is a article by BBC in 2014 stating that Nigerians eat its shoe leather. And there was a rivalry act noticed by BBC in 2015 by Nigerians, which says, bring back Pomo. So you may be wondering why beef hides. So most popularly, everyone knows that beef hides or beef hides or the originally originated from slaughter facilities, and they are available for cheaper price. So here you can see a woman selling pomo in Nigeria. Coming to manufacturing methods, in early January 2020, the first time in United States, pomo processing was started in FAPC at Oklahoma State University. So the processing methods, the manufacturing methods are trade secrets, which I cannot tell in this presentation. The main objective of this study, before that, we went through the process. Me and my advisor went through the process and identified two main crucial steps in manufacturing edible beef skin. One is dehairing and one is singeing. So Dr. Bowser came up with a scorching experiment where we employed on different methods like hair-on, trim, skin, and chemical dehairing treatments. And we also analyzed the quality parameters like color, texture, moisture, and we also had an expert sensory analysis. So coming to the moisture content was analyzed using a gravity event method mentioned in AOAC official methods. Higher moisture content in hair-on, which is 66.2, was observed, and the lower moisture in trimmer was observed as 59.28. So the higher the moisture result in the higher the yields. In general terms, if a food product has high moisture, generally the customer is buying water. So the hair-on shows more moisture level than rest of the treatments. So we analyzed color using hedonic scale sensory analysis and Hunter calorimeter. So here you can see on we made an one-way analysis for L treatment and A treatment and B treatment. We made like color versus treatment size. The hair-on treatment got more L colors when compared to the rest of treatments. And the chemical dehairing got more A values, which is red and green values when compared to other treatments. We made a hedonic scale sensory analysis on a scale of 1 to 9, where we asked the questions on general appearance and golden color. So 3 processors who make pomo every Friday in FAPC were examined with these questions. Which samples have the best golden brown color, and which sample do you prefer based on general appearance? So the chemical dehairing samples got a score of 8 out of 9, where the rest of the hair-on, skinner, and trimmer treatment got less values when compared to chemical dehairing values. The Hunter L values and the general appearance values are significantly different, and there is no correlation existed between them. And L star and A star the means are significantly different to the golden color values as well. There is no positive correlation between them. So texture, we measure texture using a low-cost texturometer to measure hardness. So here, the hair-on samples have the more hardness, and the means were examined to check if there is any significance between the samples. So here, the red circles you can see, the 4 treatments means are significantly different. And a hedonic scale sensory analysis was made on 1 to 9 scale, and the same processors who make pomo on every Friday in FAPC were examined like how hard is the sample. So then the chemical dehairing, they liked how hard is the sample. They said like they liked chemical dehairing more than hair-on or skinner and trimmer, and they gave a score of 8 out of 9. We try to analyze the differences between. We try to analyze if there is any significant correlation between the texturometer hardness and hedonic scale hardness, but there is no correlation existed between these two different types of methods. So I also analyzed time versus temperature, where the hair-on treatment took a long time to scotch the sample at 219 degrees Fahrenheit for about 4.5 minutes. The trimmer and skinner treatment took less time when compared to hair-on, and they squash the samples at 181 degrees and 180 degrees Fahrenheit for about 1.5 minutes. Just like the heron, the chemical deherring treatment took like an average of 2.5 minutes to squash the sample at 205. Here is the POMO, but you can see. I'm ready to answer the questions if you have any. Thank you.
2 minutes for questions.
Yeah, Sriram, I guess, there was a question posed to the Biltong presenters about, is there a market for that and would it be acceptable? Even though that is similar looking, even a little different than beef jerky, this product is a bit different than most anything else in the US, but I understand you can't make this fast enough to satisfy the demand for it. Is that true?
According to my understanding and the reviews that I got from the outline, so there are many Nigerians who like POMO, but there is a big black market POMO going on in every store, in every Nigerian store.
Oh, okay.
But now we replaced mostly our client is focusing on to replace the black market with the USDA label POMO.
Let me ask you. I've seen some pork processing where I've seen some big slaughter facilities and processors where the pigs look so clean, they look like they're made out of plastic, and there's no hair on them. So, but in order to use the beef carcasses, you got to do the dehairing.
Yes.
Is there a problem with using, say, pork hides as opposed to the beef?
So according to our client, we didn't examine about the pork hides because there is an existing product already called pork rinds in the market. But most probably, my client is focused on these beef hides because in Nigeria, the beef hides are so cheap and they are readily available to pick and make POMO.
And that's time.
Okay. And with that, thank you very much, Sriram.
Thank you.
And our last of the grad students is going to be, next to last. No, we got two more, excuse me. Srijana Panta, close enough.
Yes.
How do you pronounce your first name?
Srijana.
Srijana, okay. And she'll be presenting on water deficit management in pecan orchards. All right, okay. That's all yours. Take it away.
Thank you. So can you see my screen?
We see it.
Thank you. So hello everyone, my name is Srijana Panta and I am a graduate student in Department of Horticulture and Landscape Architecture, and my major advisor is Dr. Lu Zhang. So for today's presentation, my topic is water deficit management in pecan. So a brief introduction to pecan. So pecan, which is also known as Carya illinosis, belongs to the family of walnut, that is Jugulardanceae, and it is one of the deciduous tree. So pecan is a highly nutritious crop as it has high fiber content, and it is native to North America. And USA is one of the top producer of the pecan, and Oklahoma is also one of the top 5 producing state of the pecan. So to study the water management in the pecan, like one should be able to know the pecan from early growth stage to the until it is harvested. So during the early June, there is a fruit set in the tree and one can see a small knot. So when we cut this small knot, we can see there is a small development of ovule in the knot. So after this stage, there is a water stage which lasts for about 1.5 months. So during the water stage, there is an elongation of the knot. So This stage is a very important stage of the knot, in which I will explain later in another slide. So after a water stage, there is a gel stage. So in the gel stage, there is a jelly-like substance in the knot. This lasts for a very short period of time, like for 1 week or 2 weeks, depending upon the variety and location. So after the gel stage, there is a cell hardening stage, or dose, which we normally call a dose stage. So during this stage, one can see the cell of the knot turns brown into color, and the outer part of the cell, which is known as a sock, so this splits. So this signifies the grower that our pecan is ready to harvest. So a brief introduction to the water stage of the knot. So as we cut the knot during the water stage of the knot, we can see that water is splitting out of the knot, and this water is known as liquid endosperm. So this liquid endosperm will determine the future of the kernel. That is, it will provide the nutrient for developing kernels. So this is the important stage in which a grower needs to provide a careful management practice like water management, insect pest management. Otherwise, it will affect the yield of their orchard. So mainly in the Oklahoma case, growers irrigate their field according to their personal knowledge, like they do not have any scientific knowledge regarding the irrigation condition, like whether they irrigate, some of them irrigate less, or some of them irrigate more than necessary, which is only increasing their cost of production. So first, what we did is that we did some background check in our Perkins orchard to check whether there is inadequate or excessive irrigation in our field. So what we did is that we placed a water marker at the depth of 30-inch and 60-inch depth, and what we found is that our pecan orchard is irrigated more than necessary, which is only increasing our cost of production. So this brings to our main objective, that is to cut off the price of irrigation by developing precise irrigation schedule. So for the method, this research was done in Perkins research station. So we selected two varieties, that is Pony and Kanza. So for one variety, we selected 3 trees and divided them in 3 types of irrigation. So for one tree, we give them a full irrigation. So full irrigation is that irrigation amount in which the people have been providing the irrigation from past. So for the half irrigation, we half the amount of the irrigation than the normal one, and for the no irrigation, we didn't provide any irrigation. So what we measure is that is steam water potential of the tree by the use of the pressure chamber. So the pressure chamber assists in irrigation management decision. And also steam water potential is also a straightforward indicator of the plant water stress. So for the result, as you can see in this figure, this represents the water potential of 2 different varieties under different irrigation condition. So the water potential for both the variety and different irrigation treatment, it was low at the beginning, but when the drought prolongs during the month of the August, the water potential reached maximum for no irrigation and also increased for other kind of treatment. But during the period of 24 August and 31 August, we received a rainfall of about 1.33 inch. So after receiving that amount of rain, so the water potential of the tree reached to its, some of them reached to its original form. So this determines us that we can use a pressure chamber for assisting in the field. So our research was conducted until the harvest. So during the harvest stage, when the knot were harvested, we collected about 50 knots for each treatment and for both the variety to compare whether there is any difference in their weight of kernel or not. So what we found is that there is an insignificant difference between 3 kinds of treatment for both the variety. So our current research revealed that the irrigation in summer facilitates the yield yield of for both the variety, but since the yield of for the half irrigation is not reduced, so in future we can suggest the farmer that we can reduce the irrigation to the half, but further research also needed to prove this phenomena, because this research is based on only 1 year research. So this concludes my presentation. Thank you. If there is any question, I would like to take it.
3 minutes for questions.
Hello, Serena. I have
Hi.
a question. Hello, it's a good presentation, and I have a
Thank you.
question regarding. So from this table, you just showing like for the weight of either the shell plus the kernel or the kernel. There are no differences between the full irrigation, half irrigation, and no irrigation. So
Yes.
does that mean growers do not need to give any irrigation to the trees, or have you tested other like the grading, other index of the gradings?
Yes. Yes, I have also tested the grading, like in the case of pony. When I compare the skin of full, half, and no irrigation, I found out that the full irrigation have more white skin than the half and no irrigation, and other quality parameters like well, which is still I need to perform that in summer. Maybe hopefully we will find some result for full, half, and no irrigation. Even though there is no weight difference, we cannot say confidently that we should not give any water to pony. I mean to the pecan, since since the research is in tree, like it doesn't show immediate response. So I think two or 3 year more research should be invested in the pecan to identify what amount of water is needed for the pecan. Thank you.
Thank you.
1 more minute for questions.
Okay, so I will continue my question. So you see, you have say the liquid stage actually is the water stage of the pecans, and that is very critical stage of the pecan, because we have the liquid endosperm. So based on that way, do you have any plan this year you will test the contents of the liquid endosperm?
Last year I have collected the liquid endosperm by a syringe. I have some liquid content, and I have planned to test some what are the nutrient present in that. So if I have time during the summer month, I will do that.
It's great. Thank you.
Thank you.
And that's time.
Okay. Well, thank you very much, Sriana.
Thank you.
Now, if they can only figure out how to make an edible product out of acorns, I know a number of yards that are completely solid floor of acorns. So I guess there's too many for the squirrels to grab. Okay, our next presenter is Srideva Pamula Venkata, and she'll be speaking on application of cinnamon oil nanoemulsion to inhibit salmonella species on alfalfa seeds. All right. Okay. Sorry if I didn't pronounce that name correctly. Okay.
Yeah, you pronounced it good. Yeah.
Oh, okay. Good. It's all yours.
Okay, I'm gonna share my screen now. Okay, good morning, everyone. And my name is Sridevi Pamula, and I'm a graduate student in University of Central Oklahoma in the Department of Nutrition, Food Science, and Microbiology. And today I'm going to talk about the effect of cinnamon oil nanoemulsion for its antimicrobial efficacy on salmonella on the alfalfa seeds. So have you ever thought of asking yourself, is my food free of microbes? Don't you think staying healthy is the most important thing for us humans, like eating the right food and exercise? So I think the maintaining gut health is having good health is the key to good health. And for that reason, I think many people, not during the pandemic, before pandemic also, people are switching to plant-based diet, and our major portion of their diet is plant-based. So I guess consumption of sprouts have increased, especially alfalfa sprouts, maybe mainly because of its health benefits. And guys, do you also know that alfalfa sprouts are also called as father of all foods, mainly because of its high mineral content, vitamin content, and it has a high protein content. And studies have also shown that alfalfa sprouts have a more beneficial effects in preventing cancers, different kinds of cancers, osteoporosis, also in menopausal symptoms. So due to this, there is increased consumption. There's also increase in production. And this was implicated in increase in foodborne illnesses linked with alfalfa sprouts associated with salmonella. So what are the conditions that favors bacterial growth? So sprouts are typically consumed raw, and microbiological analysis showed that it has a large amount of microflora. So not just that, there are several possible pathways where you can, the alfalfa can be contaminated with salmonella from farming, harvesting, processing, maybe sprouting, transportation also. So of the many sources, I think the sprouting is the major source of contamination, because sprouting is done in warm and moist environment, and which contribute to the rapid growth of bacteria. And everybody knows that FDA has sprouting guidelines where you can use organic chemicals and their salts to prevent bacterial growth, for disinfect the sprouts. So the main objective of my study is to use natural antimicrobial agents to prevent bacterial growth. And I'm using alfalfa seeds as my major study on to check its I'm using nano emulsion to check its efficacy on the alfalfa seeds. And also, I'm also checking how it affects the germination process. So, why do you want to use essential oils? We know that cinnamon oil is an oldest and valuable spice which was used continuously in traditional Ayurveda, Chinese medicine from thousands of years. And essential oils, the cinnamon essential oil, which I'm using in this experiment, is extracted from the bark of cinnamon or from the leaves. So it is used as spice, herb, and it has diverse microbiological properties, antimicrobiological, anti-inflammatory properties, and anti-cancer properties. And it is also used to treat many other physical ailments. And it can also be used in toothache in the form of eugenol. So we know that nano emulsions have more antimicrobial properties, and but the nano emulsion essential oils have some drawbacks. That is the reason we need to formulate a nano emulsion using ultrasonication method. As you can see on the screen, where this is ultrasonication machine. So I formulated ultrasonication method, nano emulsion using ultrasonication method with a predetermined concentration of the tween 80 DI water and cinnamon oil at 60 amplitudes. And I also checked the nano emulsion stability using centrifugation method because now cinnamon oil is thermodynamically unstable. And the other drawbacks of the cinnamon oil, it has low water solubility, low stability, and it has strong sensory composition. That is the reason it is not used in the food system because of that. So I think preparing nano emulsion would be a viable option for to use it in the food system. So when I checked the stability, I saw that there was no separation or creaming observed even after 60 days of period of standing. So this stability was checked at 0 hour, 0 days, 30 days, 45 days, and 60 days. So MIC and MBC. So since we have the nano emulsion, we have to check the antimicrobial efficacy of the nano emulsion we prepared. So minimum inhibitory and bacterial concentration. So MIC is used to check the lowest concentration where the nano emulsion is capable of inhibiting the bacterial growth, and MBC is at what concentration the nano emulsion can kill the bacteria. So for this purpose, I used microdilution method and nano emulsion and the inoculum, in this case, Salmonella. I used 3 different strains of Salmonella for this experiment. So Salmonella was serially diluted in a 96 well plate. As you can see in the picture, I have a different color combinations because of the growth of the bacteria. There's a lighter color because of the growth of the bacteria, and the blue color is because there is no growth. Okay, and all the 3 different strains showed MIC at 0.039% and MBC at 0.078%. So for the method I used for this experiment is fresh bacterial culture is made using all 3 strains, and I prepared a cocktail out of it and incubated in phosphate buffering solution after centrifugation for 4,000 RPM for 20 minutes. And the seeds are artificially inoculated with the cocktail for 24 hours, and then the seeds are dried and treated with the nano emulsion for 60 seconds, and again they are dried for 30 minutes. And then I did the homogenization after drying the seeds and pounding them into a smooth paste, and the homogenized extract is extracted, and it is serially diluted for enumeration. And I did the enumeration at 0 hour, 24 hour, 48 hour
2
on
minutes remaining.
XLD plate agar, and Conleys are counted to check the efficacy of the cinnamon oil nano emulsion. Control group is also done using the same procedure. And I use distilled water as a control, and all the experiments are done in a biosafety cell hood using under UV light, and all the experiments are done in triplicates. So for the results, I saw that there is a log reduction in 0.75% and 1%, because I use the concentration of 0.75%, 1%, and 1.5% of nano emulsion in my experiment. When compared to the distilled water, and 0.75% showed 1.16 log reduction, and the 1% showed 1.32 log reduction, and the greatest log reduction was seen at 24-hour interval. The results also showed there is no interference in the germination process, and 90% of the seeds have germinated without any significant difference in the depth, in the length of the sprouts. And this research is funded by USDA and NIFA, which is awarded to the University of Central Oklahoma. You can see the references on the screen. And any questions? Thank you.
30 seconds for questions.
Sridevi, curious, you used XLD for enumeration?
Mm-hmm.
Did you ever compare that compared to like a non-selective media like TSA? Because typically, XLD is used for detection, not enumeration. And we've noticed you get reductions of salmonella even on that type of media.
Mm-hmm. I haven't tried that, Dr. Peters, but I would consider doing that in my next experiment using I'm working on the sprouts for now for after the seeds, so I would consider doing that. Yeah.
Okay.
And that's time.
All right. Okay. And we're a little bit long. Chuck, is Jason around?
Yes,
There,
he's
there he
here.
is.
There he is.
Okay.
I'm here, guys.
All right.
Good morning.
Morning.
Amazing.
How y'all doing?
Are you in sunny Florida?
No, I'm actually in Kansas City.
Oh, okay.
In my home office here today. So.
All right, that's right, you are. Well, you know, we tried. I guess a third time is a charm. We tried three. This is our third time we tried to get you here, and ice storm knocked us out. COVID knocked us out, and now we're ready. So with that.
Yep.
Go ahead.
Nope, go ahead.
And so we're happy to present Jason Reicks from Florida Food Products Incorporated. He does work with food ingredients in relation to natural curing of meats. We've actually used their product for some of our hot dogs that we've made, the vegetable celery nitrite. And so I suppose you're going to be talking a little bit about that at least, or covering that. And he's been in sales and marketing, and also in R&D in this area. And you might want to just describe a little bit about Florida Food Products and you know, what type of, how they're positioned in the market. So with that, I'll hand it over to Jason as our keynote speaker today. So it's all yours. I know we're running a little bit late, but you got the full hour, so take it.
Okay.
Okay.
Well, very good. Well, thank you, Peter. Certainly appreciate the opportunity today. As you mentioned, you know, we've planned this a couple of years back, and had some unfortunate circumstances the last couple of years with the ice storm 2 years ago, and then the pandemic last year. So really appreciate the opportunity today, and hope I can you know, share some valuable insights into natural curing in the natural and organic meat space. I know you know, there's a lot of you know, research and developments being done in this area, and it continues to be refined. So with that, I'll kick it off a little bit. I won't go too deep into detail regarding Florida Food Products, but really we're a major player in the natural food ingredient space, developing and delivering various replacement to synthetic chemicals. The big one really obviously is the replacement of sodium nitrite via vegetable plant-based sources like celery or Swiss chard. And then also varying complement products that aid the curing process, like acerola, cherry powder, and some various other items. Continuing down that path, I won't talk much about it, but looking at various other ingredient platforms, including antimicrobials, as well as one recent launch replacement of sodium phosphate, with our Veg Stable Plus. So we're really excited about that product as we continue the journey towards all natural and clean label meat products. So with that, today, what we'll be discussing is essentially really the holistic view as it relates to natural curing. So we'll go deep into you know, what is sodium nitrite, you know, the background of sodium nitrite, what it's used for, in that. Oops, sorry about that. Sodium nitrite, we'll talk about the natural curing and as it relates to vegetable processing and how do we get to that natural nitrite source. We'll talk about the regulatory and food safety aspects of natural curing, as well as bring it full circle in terms of what it means for the marketing groups at these various meat companies, as well as you know, what the consumers are demanding from a you know, a meat purchase standpoint. terms of the purpose of sodium nitrite, so again, many of you have, worked with various, different types of sodium nitrite, be it a Prague powder or various other synthetic versions of a sodium nitrite or nitrate used for curing various types of meats. So nitrite is added to meat to essentially provide a flavor, color, as well as a texture. So again, it's being used in the industry for all of those attributes as well as many more. Preservative attributes is by far probably the most important as it relates to protecting the meat from spoilage bacteria like Clostridium botulinum and various other bacterias, as well as it has a very high antioxidant property in terms of controlling various oxidation, including fat oxidation. In terms of the curing reaction, many of you have probably studied this or seen this in various, classes that you guys are going through. Essentially what's happening in the curing reaction is the nitric oxide is interacting with oxygen and converting the oxygen myoglobin to nitrose myoglobin. And then when you heat that, you're creating nitrose hemochrome, which, in this example, it turns the ham pink, which is what consumers desire. With a curing reaction from nitrite to nitric oxide, so when you use a standardized curing agent like a synthetic sodium nitrite, this is the pathway in terms of the nitrite to nitric oxide. In the case of a natural source of nitrite, we're actually taking the crude form, which is the nitrate, and we're actually converting the nitrate into nitrite versus a microbiological fermentation process that reduces the nitrate to the nitrite molecule, and then it follows the pathway to nitrous acid via an acidic environment, which various curing accelerators can promote acceleration in that process. So again, that's just the background of nitrite and what it's used for in standard in the meat industry. In terms of natural sources of nitrite, this is the pathway that this would follow. So, if you were to come to our facility, you would see various trailer loads of celery. So that process is essentially goes through the product is unloaded. It goes through a wash and grading process where then it is grinded and blanched, and essentially it's juiced, where the juice is separated from the organic matter, the biomass. So the fiber is extracted from the juice essentially. So in this case, to create natural cure or natural nitrate or nitrite, the juice portion is only used. The byproduct, the celery fiber, in this case, would be used in cattle feed, which is supporting the local farmers in Florida. So that's where the biomass goes. In terms of the key step here, as I mentioned, celery is a very high nitrate containing vegetable. There's various different levels within various celeries, although converting that nitrate into nitrite is a very important process within our manufacturing process, and that's the fermentation step, or you can call it the conversion step, or even the culturing step. So that's a very important process that is highly monitored via pH, agitation, and really time as that conversion happens from nitrate to nitrite. There is a special bacteria strains that are used in that fermentation process. The most desired one is a Staph carnosus bacteria, where the nitrate is reduced to nitrite, which is the active ingredient for curing meats. So after the fermentation or the culturing step, the product goes through a pasteurization step where, you know, it essentially kills all the leftover bacteria that's in the juice. Then the juice is evaporated and pasteurized again, and then dried into a fine powder where it would be used in a dry format for the meat industry. So a very complex process, starting all the way from, you know, really, you know, the raw celery all the way through to the final powder that's used heavily in the meat industry. So in terms of the processing, as I mentioned, you know, the celery here, you see a field here in South Florida. You know, the celery is a very compact vegetable. So there's you know, in terms of economics, it's a very good vegetable for this process, because you can grow a lot of celery per acre of land versus various other you know, vegetables that are more petiole based, and they have a wider you know, land usage from that standpoint. So again, celery, you know, it's grinded, separated, fermented, concentrated, and dried. So in terms of the celery powder, so there's really two different versions you could take a look at. I would say when the technology started roughly you know, 15, 20 years ago, it was really more about how do we you know, standardize the nitrates in the various vegetables, and then the meat processors would actually do that micro fermentation within the meat in situ within the meat matrix, and that posed a lot of challenges in terms of you know, residual levels, you know, with you know, how much was actually converted into nitrite and that sort of thing. So that's when the process was essentially brought in-house and doing that you know, within a large manufacturing facility and large tanks to really optimize and standardize that active ingredient. So, you know, the nitrate version, and then obviously the sodium nitrite version is instant curing. So it's you know, it's already converted from nitrate to nitrite. And that's how most of the celery powders are sold today in the market, using the pre-converted versions to you know, get the best performance. As I mentioned earlier, the cherry powder. So cherry powder is a natural source of ascorbic acid. So it's used in the curing process as a accelerator. So it's essentially accelerating the reaction of the curing reaction. So also, you know, it delivers various other functions within stabilizing the nice cured pink color, as well as you know, has a very high oxygen scavenging capability, by where it can improve the shelf stability of various meat products, as well as the food safety benefit, which I'll go into a little bit deeper in the next few slides. But it's essentially a hurdle for Clostridium perfringens, and now is recognized for the Appendix B from the FSIS and USDA. One might ask, you know, how does it work versus synthetic nitrites? The product is you know, standardized, so you can do your calculations to dose it at traditional levels. So your efficacy is really the same in terms of food safety as well as product quality attributes. There are some minor processing things to keep in mind as you work with the natural cures. There's been a lot of work done in this area the last 10 to 12 years, and most of the major processors, have all of this figured out in terms of how do you optimize the process in terms of maximizing the product use and using the natural cure. So again, there's in any curing, distribution is key. So to get the ingredients distributed through injection processes, tumbling, mixing is a critical step for processing to maximize the distribution to get the nitrite dispersed properly. Maceration, tumbling, increases that distribution. And then obviously, with holding the meat blocks or holding the mussels prior to cooking is a standard practice in the industry that allows the curing reaction more time to essentially convert that nitrite to nitric oxide. Here's an example of a typical formulation. So again, if you look at the chart below, there's a traditional ham formulation that would have the standard ingredients like sodium nitrite, which is a curing agent. Obviously, the sodium erythorbate, which is your curing accelerator, and then various other sweeteners as well as salt. The formulation on the right is actually an example of a natural ham formulation where all of the artificial or synthetic ingredients are removed and natural inputs are substituted there. So sea salt, turbinado sugar, the celery powder, and the cherry powder. So again, very, very similar formulations as it relates to the usage rates and that sort of thing, but it's this is an example of one example of a natural ham versus a traditional type formulation. So what does that mean in terms of product labeling? This is representative of say a traditional natural product versus the traditional counterparts. So on the top you'll see very few words. So again, a lot of, consumer understandable ingredients that you might have, in your pantry at home. So celery powder, cherry powder, citrus powders, those types of ingredients versus the synthetic counterparts. So again, this is, one of the main drivers of, natural meats and cleaner labels is really to clean up these ingredient statements, remove the synthetic chemicals, but also shorten the ingredient declarations. So on the bottom, you'll see, a lot more, chemical type additives like MSG, sodium nitrite, various, hydrolyzed corn and soy proteins, those types of ingredients, or even on the traditional Franks, there's different types of antimicrobials that would be used to preserve shelf life versus, like in the top, there's different vinegars that are used in replacement of those diacetates and lactates. Again, here's just another example of what I mentioned here. So cleaner labels on the right, that's a natural product. So there's the the cultured celery and the cherry powders. In terms of regulatory, again, this is a key area, and this area could be changing here in the coming years. But you know, the FSIS recommends, you know, 100 ppm minimum sodium nitrite for processed meats. And again, that's really around the efficacy for CBOT control, as well as, you know, food safety and, you know, really product quality as it relates to, you know, creating a nice consumer appealing cured product, a nice pink color. So I mentioned earlier about the Appendix B. Appendix B is essentially the cooling standards for these meat products. So there is, back in 2016, late 2016, early 2017, there was essentially option 3 was revised to where, you know, if you wanted to follow that so-called cured cooling schedule, you could do that using natural sources of nitrite with the addition of a natural accelerator like cherry powder. So that process is in place, and it allows a little more flexibility to the processor as it relates to cooling parameters. In Canada, again, that's a different regulatory body, so the CFIA. So their regulation is essentially 100 to 200 minimum sodium nitrite for any alternatively or naturally cured product, either manufactured in Canada or if it's manufactured here in the US and then shipped to Canada. So that's their regulation for natural sources of sodium nitrite. Again, in terms of food safety, you know, in order to maximize shelf life, you know, traditional levels should be used. Celery nitrite is really no different than sodium nitrite. So if you want the similar results in terms of food safety, you should dose accordingly and adjust your ingoing rates to get the same ingoing ppms. So in this example, it would be, you know, for a comminuted meat product like a hot dog, you would have, or a sausage, you would have 156 parts per million ingoing of the natural source of nitrite. And then there's the regulatory level for ascorbic acid, which is 469 parts per million for ingoing on the cherry powder. So that's essentially the natural curing system. They work hand in hand synergistically. In terms of labeling claims, you'll see, you know, as you shop through the supermarkets, you'll see various, you know, different labeling claims used in the industry. So really, it starts with the all natural, and that seems to be the big overarching halo of the claims. And all natural, again, there is a definition for all natural as it relates to ingredients. So you can't use any synthetic ingredients like sodium nitrite or sodium phosphate, or you know, the various antimicrobial ingredients like lactates and diacetates. You would need to incorporate substitutions to those synthetic chemicals. So that's the major the difference and the requirement to label all natural. In order to you know, use natural cure celery powder in these various meats that do contain a standard of identity of being cured, like bacon or hot dogs or ham, there's additional requirements needed on the label, like no nitrates or nitrites added, except for those naturally occurring in cultured celery powder, as well as you know, no artificial and synthetic preservatives, or keep refrigerated under 40 degrees. So those are some of the other requirements in order to utilize these natural curing components as the regulation stands today. So I talked a little bit earlier on the natural definition. So again, it's the product cannot contain any artificial flavor or flavoring, coloring, chemical preservative, or any other artificial synthetic ingredient, as I mentioned, like sodium phosphate or sodium nitrite. The products must be minimally processed. So the processing, as I discussed earlier with the celery process, is considered natural because it's a minimally processed processing methods. It's extraction via physical extraction through the juicing process is considered minimally processed. Fermentation or culturing is considered minimally processed. So there's standards that you need to meet in order to be an all natural ingredient that then goes into the all natural meat products. Okay. In terms of using the natural sources of nitrite, again, as I stated earlier, there's really three things that you must label per the CFR 9 CFR 317.17. So in the case of a beef hot dog or a beef frank, you're required to label on the front banner uncured. So that means essentially that product does not contain standardized synthetic sodium nitrite. Sodium nitrite is deemed a curing agent. So any product that is, has a standard of identity for being cured needs to use a curing agent. If you do not, then you're required to label it as uncured. There's a lot of discussions going on right now with the FSIS USDA on that, and there will most likely be some changes to that, removing the uncured to really eliminate some confusion within the industry as well as at the consumer level, because ultimately these products are cured. They're just cured naturally versus synthetically. So again, there's some possible changes coming down the pipe, in the next probably next few years on that to clear up some consumer consumer confidence in that. So no nitrates or nitrites added except for those naturally occurring. As I mentioned, that statement is required when utilizing natural sources of nitrite, as well as not preserved, can keep refrigerated below 40 at all times on that. In terms of the standard of identity, as I mentioned, the Franks traditionally cured a labeling banner. In the case of something that doesn't have a standard of identity for being cured, like a sausage or beef jerky or like a snack stick, those products are not required as the regulation stands today of being uncured. So that's why you might see, a beef jerky that's utilizing celery powder, but the product is not labeled uncured beef jerky. Okay. In terms of organic, you might wonder, can you use natural cure process in organic? Yes, you can. The NOP, the Natural Organic Program, is essentially has a list of ingredients that are essentially grandfathered in. They are on the allowed non-organic list. Celery powder is one of those ingredients, for the reason being that the amount of celery, organic celery needed to support this type of market is very large. The majority of the organic celery is used for the fresh market and retail. So there's really not a significant outlet of additional, acreage for organic celery. So that obviously can change as more farms convert to organic farming, but there's certainly a long time frame for that conversion to happen. It can take anywhere from 3 to 5 years to convert a parcel of land from conventional to organic. So as it sits today, the standard celery powder, since it's used at such a low level within an organic meat product, the standard version can be utilized in these organic meat products. Okay. One might ask, why use, natural cure? It's essentially still sodium nitrite. What is really driving the movement of, natural ingredients versus synthetic ingredients? And really, at the end of the day, what it boils down to is what the consumers are demanding. Consumers are demanding cleaner ingredient statements, shorter, recognizable ingredients, and with that, there's really more people consuming those types of products versus the traditional versions that would contain sodium nitrite. So we actually did some marketing research as it relates to synthetic versus natural ingredients, and consumers are deterred to buy products that contain chemical ingredients. So here's just a quick snapshot of some of the findings that we found, and essentially 50, about half of the respondents were would not buy a product because it contains sodium nitrite. So that's a huge half of the respondents here. So that's where a lot of people are really eating healthier, or really looking for that cleaner ingredient statement as it relates to these various processed meats. In terms of new product launches, if you look at the the last several years here, the snapshot of various products, new product launches in the retail markets illustrates that the use of celery is increasing and the use of sodium nitrite is decreasing. So if you look at the major movement really happened in 2017, 2018. That's when major companies like Oscar Mayer, Hillshire, many of the other private label brands, that's when they converted majority of their products to utilize the natural sources of sodium nitrite. Again, taking it full circle, looking at, natural brands versus traditional meat brands. So again, here there's a significant growth factor. The natural brands are growing at a faster pace than some of the traditional brands. That's why you see, a lot of the major companies developing natural brands, or you see a lot more of the natural brands, popping up as those products start to really perform better in the marketplace. Again, this is all retail based. Again, this is also happening on the food service side of the business. So again, there's a major movement here as it relates to cleaning up, labels. So back in 2015, Panera Bread, they essentially developed a internal no-no list, they called it. So essentially prohibiting the use of various synthetic chemicals in their products. And again, this goes across all of their products, including bread, meats, their soups, all of those products that they carry. So again, here you see some of the major ingredients used in processed meats like ham and bacon. Nitrates, nitrites are on the list. Sodium phosphates are on that list. So essentially, their processors and their suppliers or vendors have developed all natural versions for Panera to meet this requirement. And again, Jimmy John's, again, another a major food service or QSR that's really headed into that natural area as well. So they've came out bold with their use of all natural meats. So again, that's another example of it not just happening in the retail markets, it's also in the food service arena as well. Continuing on that, so at retail, I mentioned the major brands have converted from the synthetic to the natural. Many of the private labels that you might see at your local grocer, like Great Value at Walmart, or various brands at like an Aldi, or you might see Kroger, HEB. All of those various retailers have their own private brands. They're heading down this path as well as it relates to eliminating the synthetic nitrates and nitrites and utilizing natural sources. And with that, I think one key takeaway here is that, the no artificial preservatives and all natural, those types of claims resonate very high with consumers. And really the anchor ingredient behind all of those claims is the natural sodium nitrite. So you can't make, an all natural claim and still use Prague powder. So it truly is the anchor ingredient for the natural meat and the natural food markets, as more consumers are demanding those natural, no artificial preservative type products. So it really allows these marketers and food manufacturers to really develop unique marketing campaigns around it could be natural, it could be organic. You think about, some of the other claims that might come with that, like, grass-fed beef, all of these various other, claims without, having the natural source of nitrite, you really wouldn't have, this whole, paradigm shift or this movement toward clean, natural meat products. So with that, I'll open it up for questions. I know I went rather quickly. I'd rather have some dialogue here if you guys have questions as it relates to natural curing or some of the regulatory positions or changes coming, or in general, if you have any questions.
Hi, Jason.
Hello.
We're here. I am Ingrid Gums-Diaz from Quantum Laboratories. We're a commercial lab in Oklahoma City, food safety lab. So I was curious in something. You had mentioned on some of these, the celery powder and the cherry and whatnot, which is great for preventing, Clostridium. What does the data say, or are they about the same when it comes to any other pathogens, or what does the regulatory say when it comes to Salmonella or Listeria? I was curious.
Well, as you know, nitrite is a very good inhibitor for Listeria as well. Mm-hmm. So again, you would see, these meat companies are incorporating celery powder, again, not just for the sensory attributes, as I mentioned, for, creating the pink color or creating that cured meat flavor. You'll see the use of celery powder, say in, deli turkey or cooked chicken breasts, really for food safety purposes, like you mentioned, the Listeria control, as well as, the CBOT control. So again, it's a tool that they're using like they would synthetic sodium nitrite in their challenge studies.
Oh, okay.
As they, validate their products for shelf stability.
All right. Very good. Thank you so much.
Yep.
All right.
Jason, there was a chat question. It's kind of hard to keep an eye on the chats too, but what is the cost difference between celery powder and Prague powder?
Okay, good question. The cost in use. So again, being it is a natural input, you would need to essentially use more in volume of the natural input. It's essentially roughly 2 to 3 times the amount of a Prague powder. So again, the cost in use would be essentially around 12 to 13 cents per pound of meat, where a Prague powder or concentrated nitrite would be in that penny range. So it's it is more expensive, but again, it does deliver all of the back end marketing opportunities in terms of natural, clean label. So what many processors or meat manufacturers or marketers are doing is essentially, they do have the added cost, although they are offsetting those costs with higher premium products in terms of selling prices, and they're actually profiting more because they're expanding their selling prices beyond that 12 or 13 cents, more like 25 to all the way up to $1 per pound more. So they actually are, like I said, profiting more when they're manufacturing these types of products.
There was one more question on the chat about the sample formulation. There did not seem to be a natural substitute for sodium phosphate. How do you control water holding without a substitute? I do believe you have that, right?
Yeah, we've recently developed a product that's basis a unique proprietary process where we're extracting some specific molecules from rice bran. Rice bran is widely used in the food industry. So we're essentially extracting a phosphorus element from those from rice bran to where we can concentrate it, and it's mimicking the effectiveness of sodium phosphate. Historically, what manufacturers had to do when removing the sodium phosphate was essentially decreasing the water content, adjusting tumbling cycles, adjusting their process, increasing their salt levels to really get the performance. But now there is some solutions around sodium phosphate replacement, which is taking hold in the industry right now.
Now we have about a minute left before we start with the students. Is there any other questions?
Jason, I have kind of a philosophical question. So it's my understanding that there's really no scientific evidence that nitrates like the celery derived version are any healthier for you than the synthetics. It seems a little disingenuous if we as scientists perpetuate this natural ingredient trend when chemically, for your health, they're no different than the synthetics. I mean, why are we placating, current fads rather than just telling the truth and saying, this nitrite is really no different than the synthetic, so why do you care? Does that make sense? I understand the marketing advantage, but it seems from a scientific standpoint where we're not solving anything.
Yeah, I agree. And there's, obviously within any product or market, you have viewpoints from various angles. So you have a scientific viewpoint, you have a marketing, viewpoint as that relates. I think that's what you're seeing with the movement, possibly away from the uncured status, going away from that again is to eliminate some of that confusion as it relates to these processed meats that are labeled as uncured when they are really cured. So there's like I said, some changes that are potentially brewing there, but in terms of overall health or the difference from that standpoint, there's really like I said, I haven't seen any research that illustrates one way or another, although it'd be really compelling for someone to really take a look at that, because natural derived ingredients tend to be safer than ingredients that are derived from a chemical factory in China. The science doesn't suggest it yet, because I don't think there's really been any work that's been done in that area.
Thanks.
Yep.
All right, well, Jason, thank you very much. And you set up my presentation later on excellently. It probably helped me shave a minute off my presentation.
There you go.
Hope you stick around for that.
Yeah.
And so I thank you very much. And you've provided us celery nitrite as well for our research. So I appreciate that. Mm-hmm. We need to get back to you a little bit later, though. And so we like to continue now with the undergraduate students. And I think the next person up is Madeline Scott. Are you around? Here she is. Or Madeline. Okay. Effects of light on non-enzymatic metmyoglobin reduction in vitro. Okay.
Well, thank you for that introduction, Dr. Mariana. Like you said, my name is Madeline Scott. I'm an undergraduate student here at Oklahoma State University, doing my research work under Dr. Ramanathan in the guidance of graduate student mentor Morgan Denzer. And today I will be presenting my research done on the effects of non-enzymatic metmyoglobin reduction in vitro. So to begin, consumers prefer to see that bright cherry red color when purchasing their meat from stores, and any discoloration seen is deemed as negative in the minds of consumers, therefore leading to a greater amount of food wastage in the end. The oxidation of oxymyoglobin, that bright cherry red pigment, causes the formation of metmyoglobin, the brown pigment often seen on the surfaces of meat. The formation of metmyoglobin can be heavily influenced by common retail settings such as light or oxygen exposure. However, meat has an inherent ability to reduce that metmyoglobin and form that bright cherry red color once again. This can be done using several different metmyoglobin reducing systems, one of which is a non-enzymatic pathway. Using a non-enzymatic pathway requires an electron donor to give an electron to an electron carrier. That electron carrier then gives an electron to metmyoglobin, reducing it back to oxymyoglobin. From previous research has been done by Denzer et al. In our lab, showing us that those inherently present electron donors and carriers can contribute to non-enzymatic metmyoglobin reduction ability. However, there have been limited studies showing us the effects of light on non-enzymatic metmyoglobin reduction. We then hypothesized that the energy raised in those retail display lights will decrease or will increase non-enzymatic metmyoglobin reduction ability. Therefore, our objective was to evaluate the effects of light and dark storage conditions on non-enzymatic metmyoglobin reduction ability in vitro. Moving to our materials and methods, equine metmyoglobin solution was made at a pH of 5.6 and combined with several different electron donors and carriers. From there, complete solutions were made using NADH and ascorbate as the electron donors and methylene blue and cytochrome C as the electron carriers. Once those complete solutions were made and pipetted into 96 well plates, each of those well plates was placed under its respective light setting, which was either under an LED lamp or in a dark storage, which in our case was a spectrophotometer. From there, readings were taken every 5 minutes for 25 minutes, with the absorbance being read at 582 nanometers. The experiment was then replicated 3 times with 6 wells per treatment and evaluated a total of 18 times per respective light setting. The data were then analyzed using the mixed procedure of SAS, with the least square means being separated using a p-diff option, and were considered significant at a p-value of less than 0.05. Moving to our results, several interesting findings were observed. To start, I'll go through figure 1 first, which has an x-axis that contains the treatments and a y-axis that contains metmyoglobin reduction, all while a greater number represents a greater amount of reduction. To begin, I'll go through NADH first, which was done with no electron carrier or EDTA, and the study showed that there was actually a significant increase in reduction under light conditions. Next, when comparing the 2 electron donors using the same electron carrier, methylene blue, along with still using EDTA, the study showed that there was also a significant increase in reduction under light conditions. However, when comparing those 2 donors directly, there was not much of a difference. Next, when considering using cytochrome C as an electron carrier, the study showed that there was actually a decrease in reduction under light conditions. And finally, when considering the pairing of NADH and cytochrome C, the study showed that there is actually little to no effect made by light on reduction. And when considering the true effects of light and dark storage conditions on reduction, figure 2 shows us that there was a significant increase in reduction in light conditions when compared to dark. And although we're unsure of the mechanisms, the differences in molecular weights could be responsible for those differences seen in reduction. The molecular weights of each of those electron donors and carriers are shown here, along with a pattern in that a larger weight paired with a smaller weight resulted in a greater amount of reduction in comparison to a pairing of 2 larger weights. Although, again, we are unsure of why this does occur. And finally, in conclusion, when considering the practical implications of this study, using electron donors and carriers in a post-harvest enhancement technology could have the potential to extend color stability. Additionally, when considering using an active packaging material coupled with the cofactors of non-enzymatic metmyoglobin reduction ability could also have the potential to reduce discoloration and increase the shelf life of several meat products. Thank you. And if you have any questions, I'm happy to answer those.
4.5 minutes for questions.
Darren Scott here. Quick question. Did you look at any other light sources other than LED, incandescent or fluorescent?
No, sir. LED was the only one that we did evaluate.
Okay. Thank you.
Over 3.5 minutes for questions.
Hi, Maddie. This is Morgan. Was there a reason that you specifically used cytochrome C?
Yes. So cytochrome C is naturally occurring within the meat and muscle. So that is why we went ahead and went with cytochrome C. And then along with methylene blue is kind of the standard when looking at reduction. So that is why we chose those 2.
Three more minutes.
Got a question. How do you see this being applied?
So we can do that in two of the ways that I mentioned. We can look at using a post-harvest enhancement technology, such as an injection. There's been previous work done in our lab where there was like a rosemary injection. And so when I used ascorbate being vitamin C, we can use that as an injection process, as well as using that in an active packaging technology, using that within the packaging itself.
Why would injection? I mean, if it's a surface phenomenon, or the visible evidence is on the surface, is that in case you subsequently cut the beef that it would be protected wherever you cut it?
Correct. So it is on the surface, but it does start within, yes.
Okay.
Still a minute and a half remaining, but we can move on if no other questions.
All right, well, good work. Thank you very much.
Thank you.
Okay. As we prepare for our next presentation from Lauren Millis, excuse me. There she is, okay. Lauren Millis will be presenting on the use of brewer's spent grain for production of high-value mushrooms. Okay.
Hello, thank you so much for that introduction. My name is Liz, and as you said, I will be presenting on brewer's spent grain. So beer is made of 4 main ingredients. Those are grain, hops, yeast, and water. And most people don't
Looks like we lost Lauren for just a second.
Oh, she's still there.
Can you hear me? Can everybody hear me?
Yes.
Yes.
Okay, perfect. I'm so sorry about that. I did not know what happened. Okay, so to continue, so BSG is typically used to feed animals or is just completely thrown out. Because we noticed that the BSG has similar nutritional components to parts of the substrate that oyster mushrooms or Pleurotus austriatus are typically grown in, we decided to test their ability to produce mushrooms. Dr. Danielle Belmer, Dr. Stephen Merrick, and myself have been growing these mushrooms in substrates with substitutions of different kinds of BSG mediums. Our objective was to determine the efficiency of using BSG as a primary substrate for growing oyster mushrooms, and we hypothesized that the mushrooms would grow with similar biological efficiency to that of the control group. So the process starts off with when we transfer stored cultures of Pleurotus austriatus to new plates of MYPS plus BCRAD agar. Then we cut pieces of the existing cultures out and place them into millet jars, which we shake up to make sure that the grain is evenly inoculated. We then split the millet jars evenly between 2 substrate bags and mix those as well to make sure that they're evenly inoculated. We wait until the bags have been completely covered in the white inoculum, which takes about a week. And then we place them into a tent, which was a standing tent for growth that is humidity controlled as well as has light controls to optimize growth. When we put them in the tent, we cut holes in the bags for oxygen exposure which enables growth of the mushrooms. And we left them in the chamber for about a month each, harvesting every time we saw a mushroom fruit. So here's some more information about the methods that we used to conduct the experiments. We staggered the treatments from 1 to 2 weeks so that we could prepare for the next treatments. And we evaluated 2 treatments at the same time, splitting the millet jars evenly between them to eliminate variables. In these experiments, we tested 3 different substrates of BSG, and those were smoked pale ale, graham cracker ale, and stilly wheat ale, alongside the control sawdust. So after we did some proportion work with both the bag sizes and the sizes of the millet jars, this is the formulations that we came up with for both of these. And you can see here the hardwood sawdust pellets is where we actually added the BSG. So this is the number for the BSG that we added instead in those different substrates. And also this gypsum, lime, and wollastonite mixture, we estimated the amount because we had only lightly dusted each jar with this amount. So we estimated that to be about a teaspoon, my bad. All right, so the fresh weight and dry weight comparisons, you can see on these graphs, there are some X's. Those represent the mean weights of all of the data. And the bars right next to them represent the median of the data. And because those are so close in all of these different medians, we can determine that the data is approximately normally distributed, which is really good for analysis. So we can also see in the first 3 medians on both graphs that the mean and median weights are around the same for the control, the smoked pale ale, and the graham cracker ale, which is really good that they're all around the same. So the height of each box represents the interquartile range, which states that around exactly 50% of the data lies between these bounds. The lines coming out of the boxes represent 1.5 times the interquartile range, which helps us determine if there are any outliers. As we see here with the graham cracker ale, that there was 1 outlier. But because there were so few other outliers, we determined that the data was really good for future analysis. So biological efficiency is defined as the ability of mushrooms to produce the necessary amount of proteins for the mushrooms to benefit the people or the animals that consume them. We calculated this by dividing the mass of the mushrooms produced by the initial mass of the substrate they were grown in. You can see here on the chart that all 3 of the control groups, as well as the smoked pale ale and the graham cracker ale groups, all performed at around the same biological efficiency, which was really awesome to see. However, we noticed that the stilly wheat was significantly lower in terms of biological efficiency. On the previous slide, you could also see that the mean weights of the stilly wheat ale were also significantly lower, as well as the production rate of the stilly wheat ale. So we also sent some dried mushrooms to a lab to be analyzed for protein content. And the results that we found were really exciting because the control was lower than both the smoked pale ale and the graham cracker ale. It was also lower than the stilly wheat because the concentration in the small weight of each mushroom was much higher. But because the biological efficiency and the production rates were so much lower for stilly wheat, this data is slightly misleading in that way. However, the smoked pale ale and the graham cracker had very similar biological efficiency, growth rates, and weights. So this information is pretty significant. So we were really excited to see that each BSG sample produced mushrooms, which means that mushrooms can be grown in BSG. The analysis we did showed that both the smoked pale ale and the graham cracker ale BSG had biological efficiency levels and growth levels that were similar to that of the control group, while we saw that stilly wheat did not have quite the same statistics. So, and we noticed that they were found to have significantly lower weight and biological efficiency. We also looked at the protein content in the samples, and that was significantly higher in the smoked pale ale and the graham cracker ale. So that was exciting. Some factors that we looked at that could be better controlled included placement of the cut in the bag when we place it into the tent, as well as standardizing the time of harvest, which we had to vary because of our schedules. So some other things that we would like to look at are what components in the BSG could inhibit the growth of mushrooms, specifically like what we saw in the stilly wheat, because it didn't produce very good mushrooms for us. We would also like to look at research for uses of the waste that we produced after we cultivated the mushrooms, as well as ways to maximize the protein content in the mushrooms. So we would like to thank Iron Monk Brewery for the BSG samples and Kasner for providing funding for this research. And if you guys have any questions, please let me know. Thank you.
45 seconds for questions.
Hi, Lauren. This is Noran Danford. Is there a specific reason that you chose oyster mushrooms?
So we actually reached out to Stephen Merrick, who had already done a lot of research on oyster mushrooms. So we decided, because there was already a lot of information about these oyster mushrooms in his lab, that if we worked with him, it would be best to just continue the research on these mushrooms.
Thank you.
10 seconds. And time.
All right. Well, thank you for that presentation. Our next speaker is Guy Taylor. Is Guy around? Hello? Is Guy Taylor around? Am I missing something here?
I do not see Guy Taylor on.
Okay. Going once, going twice. I don't know, is Felicia available?
Yes.
Chuck, what would you suggest?
Well, if Guy's not here, let's go ahead and proceed with Felicia. If Guy shows up, maybe had technical difficulties, we can give him a shot later. Otherwise, we'll probably 10 ahead of schedule.
Okay. All right. Is that all right, Felicia? Are you ready?
I'm ready.
All right. So our next presenter is Felicia Johnson, and she's going to be presenting on biodiversity of the functional genomics and microbiome of blood as a response to infection in goats by the nematode Haemonchus contortus. Basically, I was contorted in trying to say that. Okay, it's all yours.
Thank you, Dr. Mariana. My name is Felicia Johnson, as he presented, and I am an undergraduate student at Langston University. And today, I'm going to talk about the research that we did with the study of alpine goat and Haemonchus contortus infection. So in the study of immune response of domestic goat, there has been an identification of blood-based results that can help determine conditions of health through the host pathogenic relationship. Haemonchus contortus is a blood-sucking parasite that can cost billions of dollars annually due to illness and death of domestic goat, Capra agagris hircus, which is the alpine goat, which we studied. The barber pole worm is the most pathogenic nematode of small ruminants. They cause anemia due to low iron and bottle jaw in domestic goats. Other symptoms noted in the host is weight loss, dysentery, leading to habitual death. The barber pole worm attaches itself to the abomasal mucosa wall of the host. And female nematodes may lay up to 10,000 eggs daily, which is a very rapid procedure. Within our research, the barber pole worm develops ways to induce depression in the immune host response. Gene expression plays a major role in response to an attack from a parasite or pathogen. And the microbiome helps modulate response to internal and external environmental stimuli. So good health versus infection of selected wethers through microbial flora and blood work is what we studied. And to the right of this slide here, this is the life cycle of the Haemonchus contortus. So the first thing the goat does is graze, and with grazing, they ingest the L3 stage larva. The next step is the adult worm attaches to the abomasum, so they grow into adult worms. The next phase is the eggs pass with feces, and the eggs develop in feces into L3 larvae in about 6 days. So that's pretty fast. And the following stage after that, the infected L3 larvae leave feces and move up to the grass blades, 2 to 3 inches in water, film, or dew. So the cycle continues. Our hypothesis was we had to ask our question, what determines immune response in domestic goat? The host genome produces an immune response due to the presence of parasites in the form of gene expression. The microbiome, what we studied, also produces an immune response. So therefore, the immune response is expressed through genes exhibited by the host genome and its microbiome to the parasites. We had 3 objectives. The first one is to analyze the different responses to different treatment types by gene expression and metagenomic analyses. The second was to identify. And what we did was identify genes expressed from the treatment days post-inoculation. And then we had to compare the microbial flora presence after 7 and 21 days post-inoculation in quantitative terms of abundance and diversity to determine the differences of health of the goat wethers. So this is a volcano plot in our data and observations. With that, in the volcano plot, it's the result of the RNA sequencing. So this identifies genes with a large fold change that are statistically significant. So in this volcano plot, there were 19 to 20 blood samples indicating likely significant expressions of genes. So the number equal to the p-value was less than 0.5 based on treatment types of the alpine goat or the Capra hircus, following star alignment with Partek flow and GSA differential analysis for transcript sequences of 7,627. Out of those 7,627, there were 523 selected. So those were the most significant or the best genes that were used from the sequence express. So to the left of the volcano plot, those were the downregulated genes that were expressed. And in the middle of the volcano plot here, those were the no change in regulation. So those were between, the number was it was equal to less than -2 up to equal to, well, they were less than, equal to, and equal to, well, they were greater than, sorry, let me read this over. They were greater than -2, equal to -2, so below -2, or below 2. And the upregulated, which is to the right, they were equal to greater than 2 in gene distribution when comparing no infection samples to infection-only samples when expressed against a numeric triad of p-values for no infection versus infection only. And they're color-coded here as well. Infection only green, and no infection versus infections, which is the zoledronic acid inject, were red, and no infection versus infection, which were the antibody inject, T-cell antibody, which are in blue. And below here, below the threshold, there are some genes that were not significant, so we didn't use those. These are relative abundance of operational taxonomic units. On the left, there's 7 days post-inoculation for 18 samples. The first number before each sample of the ID numbers and the type of treatment. So number 1 stands for no infection. Number 2, infection only. 3, infection of zoledronic acid inject. And 4 is injection of antibody inject. And to the right, these are the relative abundance of OTUs on day 21 post-inoculation, or 20 samples. So the first number again is for the sample ID, identifying the treatment type, and it has the number 1, 2, 3, or 4 for the type of infection.
2 minutes remaining.
In conclusion, through gene expression and microbial flora structure during infection with the barber poleworm, examination presents there are significant differences in the gene expression effects. The abundance and diversity of the group of organisms studied increased as the subject is older and exposed to the environment longer. So it changes as they're exposed to the environment. So this implies that Humongous contortus does effectively change and disrupt the internal habitat health of the subject host. Firmicutes and bacteroidetes, or the FB ratio of the microbial flora, can indicate host health status of our subjects. High firmicutes and low bacteroidetes suggest microbial imbalance that implies impaired insulin sensitivity, causing a dysregulation of immunity and increased inflammation. Low FB ratios indicate decreased diversity of the microbiome, also indicating an unhealthy condition. Healthy responses would be at an equilibrium state or a balanced state and would indicate a healthy alpine goat without infection to Humongous contortus. I do want to acknowledge my advisors, Drs. Jessica Kiata Pinango and Dr. Jonathan Tillahan, as well as the Evans Allen Program Grant at Lansing University.
30 seconds for a question.
Any questions for Felicia? I got one that everyone is interested to hear. Why is it called the barber pole worm?
Well, the barber poleworm, it's because of how it looks and how it wraps around itself. So.
Okay. Any other questions? Quickly, going once, going twice. All right. Well, thank you very much, Felicia.
Thank you.
Well, let me get back. Is Guy Taylor in the house? Okay, we're still missing Guy. All right, our next presenter, Rawakarn. Are you here? Okay. Rawakarn Woodarat, and she'll be presenting on functional bread, effect of red kidney bean and peanut flour on dough and bread characteristics. So with that, it's all yours.
Thank you for the introduction, Dr. Morena. Good morning. I am Ravikan from Metro University, Thailand. I was in FEPC last winter under Dr. Rayas and Dr. Pawali's supervision. Today, I am presenting results from a research study conducted with my colleague Suganda in Dr. Rayas' lab. The study described dough and bread properties from a composite mix of wheat, red kidney bean, and peanut flours. Worldwide, there is increased interest in wellness and the direct relationship of dietary pattern with health. Consumer and manufacturer would benefit from ingredients that can support health benefit and can be incorporated in consumption bread. Since 2016, there has been an effort to increase awareness of the value of pulses in human diet. Red kidney beans and peanut have high protein, about 24%, and fiber, about 25%, and thus have potential value to use in functional breads. The objective of this study is to evaluate the effect of red kidney bean and peanut flours on the physical chemical properties of dough and bread. The selection of RKB and P is part of ongoing research of design and processing pre-cooked instant flours from pulses to increase their functionality in food products. Flours were selected from previous processing experience using RKB, but for the first time, we were combining it with peanuts. In preliminary analysis, we determined the processing condition of the flour prepared in the lab by using gas oven at 325 Fahrenheit for 3 minutes, as well as the levels of each flour in bread formulation. The selection criteria were flavor, aroma, and appearance. The selected levels in composite flour were expressed as ratios. Control consistent in 100% wheat, commercial bread flour with RKB and P. To ratio of RKB flour 25% and 30%, and to ratio of P flour 10 and 15%. Thus 25 to 10 ratio represent 25% RKB, 10% P, and wheat flour 65%, while 25 to 15 ratio represent 25% RKB, 15% P, and wheat flour 16%, and so forth. The doughs were prepared and their fundamental viscoelastic properties analyzed, as well as their performance in bread making, measuring the list of key parameters of quality. The substitution of 30% RKB flower describe dough deformation up to 57% compared to control. Thus, the dough was stiffer after substitution. The possible explanation is the RKB flower that increased starch component in dough system. Thus, the non-gluten components could improve stiffness on the dough. RKB with pea flour increased dough deformation up to 50% compared to substitution only RKB flour. Dough deformation could be contributed from fats as a component in peanut flours. For bread properties, the results show that adding 30% red kidney bean flour increased protein 24% and fiber up to 66% compared to control. However, adding peanut flour reduced bread volume compared to red kidney bean flours. It could be due to a big size of peanut flour based on an observation from flour processing prepared in the laboratory. RKB flours increase darkness up to 9% and redness of breadcrumb due to color of RKB flours. Substituting RKB and pea flours in wheat flour base increase moisture of breadcrumb up to 10% and crumb ferment up to 62%. RKB and pea flours has high in amylose content which lead to more retrogradation. This review in an increasing of crumb firmness. RKB flour at 30% substitution in this bread had overall an aroma comparable sensory characteristic as with flour bread. Pea flour improved dough viscoelastic properties but no sensory score. Thus, the overall acceptable characteristic of the bread can be contributed mainly for aroma and dough deformation. In conclusion, RKB flour at 30% had overall comparable sensory characteristic as white flours bread. RKB flour at 30% improved protein and fiber up to 23.8 and 66% compared to control. Pea flour improved dough viscoelastic property but not sensory score. Future work optimize peanut roasting to improve potential use and flour enrichment in bread products. Thank you.
2 minutes for questions.
Hi, this is Kristen. On one of your slides, you said that it had, I think, a 9% increase in darkness and redness. For someone without a food science background, what does that, are there any implications or how does that affect the product?
Nine % is red. Is a red color? Yes.
Yes, and the darkness and redness, I think it said.
Yes.
Does that do anything to the bread, the taste or the quality?
Improves protein. Improve protein.
Okay. Okay. Improve protein. Thank you.
40 more seconds for questions.
Any other questions? Well, if not, then we'll continue. Thank you very much, Robert Karn. And we'll continue to the next speaker. Is it Kristen McKay?
Yes.
Oh, hey, how are you? Okay. And Kristen is going to be presenting on the prevalence of selected pathogens in ectoparasites from feral cats of central Oklahoma. She has to get out, right?
Yes, it won't let me share at the same time.
Rawakarn, can you unshare?
There's a stop sharing button.
Oh.
For
Do
her.
I have that?
No. There we go.
Okay.
Okay. So thank you for the introduction. As he said, my name is Kristen McKay, and through my research in the entomology and plant pathology department, I will be presenting on the prevalence of selected pathogens and ectoparasites from feral cats of central Oklahoma. So the implication of this study and the justification for it is that ectoparasites of feral cats have implications for human and domestic animal health. These ectoparasites are known to carry zoonotic diseases, and the close proximity of some of these feral cat populations to the residents can cause crossover between the feral cats and wildlife populations to human and domestic animal health. So a previous ectoparasite prevalence study had been conducted by professors at the vet school on this same population. So we wanted to compare our findings to their findings, but we also wanted to expand the current knowledge by testing these ectoparasites for pathogens. And we selected these pathogens based on the literature, and we decided on Rickettsia species and Bartonella species. For the methods, our sample population was the feral cats that came through the Operation Catnip Stillwater Clinic. This is a trap, neuter, and release program put on by the vet school once a month during the school year. Due to COVID, we were only able to sample in January and February of 2020, but we had plans to sample the whole year to get a full understanding of the prevalence. And to ensure random sampling, every fourth cat was combed for fleas and ticks, and we also swabbed the ears for ear mites. These collections were covered by the IOCOOK, as well as a release form that the caretakers filled out whenever they brought the trapped cats into the clinic. On the right, you can see a map that I built, and this is showing where all the feral cats were trapped from. And this is a sample size of 57 cats that we sampled in January and February. So you can see that we got a pretty good spread of central and northern Oklahoma. In the lab, we first identified the ectoparasites using keys. We then extracted the DNA of these ectoparasites using a published protocol. And through PCR, we were able to amplify the genes of interest. For Rickettsia species, it was the GLTA primer, and for Bartonella species, it was the BHCS primer. As you can see on the right, I then visualized this information with gel electrophoresis, and we were able to cut out and purify these bands using a kit, and these purifications were then sequenced by the OSU DNA lab to tell us what pathogens our ectoparasites may be holding. So the first results would be for the ectoparasites prevalence. We found that 23% of the cats sampled were infested with fleas, 16% of the cats sampled were infested with ticks, and 21% of the cats sampled were infested with ear mites. The only flea species that was identified was the cat flea that you can see on the right, and they also all happen to be female. The two tick species that we found were Amblyomma americanum and Ixodes scapularis, and then the only mite species that was identified was Otodectus cynotus, which is the common ear mite. Again, we wanted to compare this to the previous study. All of these species were found in the previous study, and the previous study also found additional species as they were able to test all year round. And then we get into the results for our pathogen prevalence. So in our fleas, we found 5 species of bacteria, 2 of which are unknown. So as you can see here on the right, this is the flea data. We found Bartonella henslei, Bartonella chlorigidae, and Rickettsia felis. All 3 of these are known to cause disease in both cats and humans. And then we also found an unknown Bartonella species and an unknown Rickettsia species. And according to the NCBI database, the previous finds of those unknown species were not found to be in cats. So you can see that we have a high prevalence rate of Rickettsia felis, which was to be expected because Rickettsia felis is thought to be a common pathogen in cats. But you can also see at the bottom, it seems to be common for Rickettsia felis to be a co-infection with other Bartonella species. And then as for the ticks, we found a 20% prevalence rate of Rickettsia felis. And for the ear mites, we found a 5.26% prevalence rate of Bartonella chlorigidae. The conclusions of this study are that we found 3 zoonotic flea pathogens in central Oklahoma. We also saw high levels of co-infection in fleas for Rickettsia felis and the different Bartonella species. So this implies that Rickettsia and Bartonella don't really seem to mind each other and they can coexist in a vector pretty easily. There was also an unknown Bartonella and an unknown Rickettsia species, which needs to be further described. As for the Rickettsia and the tick, we wonder if this particular tick had a blood meal that implies an infected cat, or if this is a new vector that we need to discover more, because Amblyomma americanum was not previously thought to be a common vector of Rickettsia felis. It is usually thought to be a flea-borne disease. This was also the first time that Bartonella chlorigidae was found in an ear mite. And in agreement with the previous study, this also shows the need for flea and tick preventative year-round, as we did still find high infestation rates in the winter months of January and February. The bottom line is going to be that feral cats serve as a reservoir for zoonotic pathogens infecting both humans and domestic cats, so they should be a population of interest whenever we think about the concept of One Health. And I do have some people to thank. First off, the funding for this project was provided by the Wentz Research Foundation. I'd also like to thank my faculty mentor, who is Dr. Bruce Noden, as well as my second reader for my thesis, who is Dr. Justin Talley. And then this picture on the right has a red arrow that shows me set up at the Operation Catnip Clinic. So I'd like to thank both Julie Allen and Dr. Kim Carter for allowing me to set up shop at their clinic and sample the cats. So I can take questions at this time.
2 minutes for questions.
Great presentation. So you identified the bacteria in the pathogen as well as in the host, right?
So in this study, we only extracted the DNA from the ectoparasites themselves. And future studies, I would recommend collecting the ectoparasite as well as taking a blood sample from the cats themselves. That was not particularly covered in this IO-Cook, but it is definitely a good research method for future people to understand the disease triangle.
Okay, thank you.
Thank you.
Another minute for questions.
So I guess the take-home point is don't try to catch and play with a feral cat then, huh?
No, unless you are bringing it to the monthly Operation Catnip Clinic and you can rent a trap from us through our website, please do not touch
Oh,
cats.
that's good. That's very good.
Yes, I've been working with Operation Catnip for the past 5 years, and you can check out their website if you see a colony and would like to help with the public health effort.
All right. Well, thank you very much. That was a good presentation. Next presentation by Sydney Cannon.
Yes.
All right. Sydney Cannon presenting on designing a knock box for the smaller packing facility. One structure for bison, cattle, swine, sheep, and goats. Okay. It's all yours.
Thank you, Dr. Mariana. Like you said, my name is Sydney Cannon, and along with my teammates Minhyok Kim and Trinity Smith, we have been designing a knockbox for a processing facility in northeastern Oklahoma.
I'm going to talk about project introduction. Our client is Tonka Processing Corporation located in Tonka, Oklahoma. This processing facility handles local processor of bison, cattle, swine, sheep, and goats. What Tonka Processing Corporation wants to do is design and build a new livestock kill chute, which have knock boxes that can restrain a variety size of livestock species.
So when we went into this design, our client had 4 main areas that they were concerned with. First of all, of course, being safety, which is how or why we switched from the pulley system with a lift gate that they currently have to a swinging gate. And then we also looked at hydraulic versus a manual system. And of course, efficiency with the USDA, that needing to be easily accessible for them to clean around and underneath the chute. And then the accessibility to the livestock's hind feet for shackling after the animal has fallen. And the biggest constraint with this is that it needed to be able to be utilized with bison, cattle, swine, sheep, and goats. And we needed to be able to do all of this under our budget of $5,000.
When we approached this problem, after considering a lot of research with current technologies within the industry, we decided to piece together different aspects of the chute, which included the head gate, the side panel, the rear gates, and the side gate. So with our head gate, we were deciding between a crash head gate as well as a catch head gate. Our side panel, whether it be tilted or straight, and if it would be either fixed or have a squeeze component. Additionally, we considered shortening the length of the chute for smaller livestock, either using multiple rear gates and multiple head gates, which would be included in the side gate. So as we moved to our final design, we decided to use a crash head gate, a tilted side panel that also has a squeeze component utilized in that, which is controlled by a slack adjuster. And we also decided to use a single rear gate, and then additional front gates were added into our side swinging gate. where the employee can go into the chute for when all the different measures are activated for smaller livestock and stun the animals through there. And as you can see in the images below, I'm highlighting the different space of the chute. On the left, you can see the larger areas for the larger livestock, cattle and bison, and potentially sows and boars. And then when all the mechanisms are activated, so the extra head gate and the squeeze component, it decreases the size of the chute for your goats, sheep, and market hogs.
I'm going to talk about what you need to do over the next 4 weeks. Before that, to tell you the current production status, the frame is completely welded and the gates and side panels are in progress. In the next 4 weeks, if there is a design failure in the fabrication process, we have to design subject to alternation. Also, we have to deliver the livestock chute to Tonkawa upon completion.
The main goal with our project, we wanted to provide a solution to a common problem. And with a lot of local processing facilities, there are, just working with local producers, they have a wide range of livestock species in a lot of different sizes, whether you have, like Trinity said, a market hog versus a sow that you're culling from your herd. But they don't always have the space for 2 different sized chutes. So our design is essentially a 2-in-1 chute that can be full size for cattle and bison and the larger hogs. And then it can be adjusted with that squeeze panel and the cutout gates from the larger side gate for the smaller hogs and then sheep and goats. And throughout this design process, we have a few people we'd like to thank, Dr. Bowser and Dr. Weckler, who have helped us with the entire process from initial concepts to the design that we're at now, as well as Joe Preston and the rest of the guys out in the BAE lab who have been helping us with fabrication and some final design changes. And of course, we would like to acknowledge our client and sponsor, Mr. Brian Lane with Tonkawa Processing Corps. So what questions can we answer?
4 minutes for questions.
So whose idea was this particular chute setup?
Well, the project was brought to us by Mr. Brian Lane, and then after a couple weeks of research and finding different technologies that we liked, we came together and decided on some of the things, and it took a little bit of trial and error to get some of the different pieces to work together in harmony, I guess. But it was definitely just a lot of trial and error between the 3 of us.
3 more minutes for questions.
Hi, this is Nurhan Danford. What is the most important thing you guys learned during this process? Obviously, your design is not complete right now, but so far, what was the most important thing you learned?
I think for me, when we went to visit to look at their current setup and the chute they had, what they were moving away from, I originally didn't know everything that went into all of the pieces of it and why it was the way it was. And as we were trying to create this design, figuring out the efficient way, the safe way, how to meet all of the requirements that it needed to. I think it would have been easier if we could have had it flat on the floor, but with cleaning requirements, it needed to be lifted up. And just learning about all of the different pieces that I didn't know about, even coming from 4 years of engineering and an agricultural background that I still didn't know were there and in that design process.
And at 45.
So you've done patent research to see what else is out there. How challenging is it to come up with a new design that hasn't already been created?
It was challenging. Our combination of the three of our ideas was also a combination of things we found in our research that we liked how they had it, how it looked like it would work for us. But we were pushed away from using other people's exact designs because there was always one thing that, maybe we couldn't make it small enough for a goat, or that head gate just wasn't right for the bison. And that made it even harder to say, well, this design would be perfect except. So how necessary is this? But being able to say this is what our client wants really helped us to move away from using someone else's design identically.
30 seconds for last question.
All right. Well, thank you very much.
Thank you.
And now we're moving on to the faculty side of presentations. Dr. Ram. So we have coming up Dr. Ranjith Ramanathan, who will be presenting on the impact of probiotics on the metabolome of cow and goat milk yogurt. All right.
Thank you, Dr. Mariana.
Okay.
Couple of years back, I was really lucky to host a scientist from NDRI, or National Dairy Research Institute from India. Those who know me, by any means, I'm not a milk person, so don't ask me any tough questions. I know this is not a competition, so Chuck, give me only 5 seconds, okay? So the overall goal was to find out how the different probiotics affect the metabolome profile of cow and goat milk yogurt. So a little bit background, in the recent years, there is a more use of, you can see more food products that contain probiotics. It's primarily because these probiotics bacteria can produce a lot of health beneficial compounds or bioactive compounds. So even though these probiotics has been the industry for a long time, there is not much studies on how these different metabolites changes with the different storage condition. So for that one, we use metabolomics. It's a really good tool to identify different metabolites present in any food matrix, and also used in a lot of medical research. When the scientists came to my lab, we were looking for a project to do. Then there are a lot of studies done in meat side, some of the cheese, but there is not much studies in the how these metabolites changes with the storage time, especially cow and goat milk. Especially goat, it's very common in developing countries. So that's why we choose cow and goat milk. And also goat, the fat size is small. It's easy to digest. So there are more health benefits with the goat milk, but there is limited studies with how the metabolite changes with the cow and goat milk. So we got cow and goat milk from the store in still water. We added both probiotic culture. We added three different types, mainly lactobacillus origin and also bifidobacterium. So we incubated in our lab condition, we incubated at 42 degrees until the pH drops down to 4.6. The metabolites were extracted. We had to play around with the methodology because there is, we started with the metabolomics approach in the meat side. Then we had to modify because of the composition and also the level of fat content. The metabolites were identified using GCMS. We use a non-targeted approach. In non-targeted, we're going to identify the entire metabolome. Whereas in targeted, we are using a specific, but here we use a non-target approach. Then the data analysis using R, and we use NIST 17 library to analyze the data. And finally, the metabolomics data were analyzed using Metabolon, that's the open source software from Canada. Coming to the results, we were able to identify 192 metabolites in both cow and goat milk. So when you have a large data set, one of the most common approach uses a PC analysis. So it's called the principal component analysis. So in this one, I'll walk through the graph so you can see two different colors. So one represent the cow, and whereas the green represent the goat milk yogurt. So this one is at day 0, so that's on the day of start after incubate, after setting in. We can clearly see both has a totally different metabolites. So the metabolite pattern in cow and goat milk are different. As we keep more time, like day 14 and day 28, again, we can see it separates out. So that means the type of metabolites formed during storage, it's different. This one is a word kind of plot. I saw a student from Langston, she presented word kind of plot. So word kind of plot, basically, it talks about how this metabolites changes. You can see two different colors. Some of them are down-regulated, whereas some of them up-regulated. And also, the colored one represents, these are the significantly different metabolites. For example, there are 20 metabolites were down-regulated, whereas 24 were up-regulated, and 148 were not significant. So this one is it's called a heat map. It really shows how these metabolites changes with, so this one is a day 14. So you can see these are the metabolites we identify. Some of them were identified, some of them were not able to identify or matches with the library. So you can see within the sample, there is more uniformity. You can see, so each color represents with upregulated or downregulated. Again, it shows within the group. So these are some of the metabolites we identify. Again, this was the first study, something reported. So we really don't know what to expect, but as expected, you can see more proteolysis. So because milk contain casein and also a lot of other protein. So what you can expect is when you're adding these probiotics, it's going to break it down. So that's why we were able to identify some of the dipeptide, polypeptide within the analysis. So some of the important or something we have more fold chain like glutamine derivative, tryptophan derivative. So all were differentially expressed. So what we did was, let's say if we have a lot of different metabolites, what does it mean? So that way we done some pathway analysis. In that one, we can see like there is a difference in fatty acid metabolism, glutathione or amino acid metabolism. This one, so this pathway analysis, you can see bigger the circle, it has more impact. Smaller the circle, it has less impact on the pathway. So what does it mean? Just to take a summary. So there is a difference. Mainly, we were able to identify amino acid peptide. That was our aim too. We want to see how much is the amino acid difference in the pattern and how the proteolysis, which one had more proteolysis or less proteolysis, that can explain difference in the acidification. So the cow milk had more proteolysis, and also at the same time, the cow milk had a greater pH than the goat milk. And also we didn't present here, we also did the fatty acid pattern. So there is a difference in the fatty acid profile that's more expected. This study shows how the probiotic culture influenced the type of metabolite form. In the long term, what is the implication? Like it can form different types of bioactive compounds. So if we can characterize the type of bioactive compounds formed in a specific set of conditions, that will help for the producers to make some science-based decision. With that, I would like to thank you for your attention. And if you have any questions, happy to answer. Again, by any means, I'm not a milk person. So if I know something, definitely happy to answer. Thank you.
2 minutes for questions.
Dr. Ramanathan, Pete here. When you say about probiotic culture, is this the culture used to make the yogurt or added to the yogurt as a supplement? Because by standard of identity, yogurt's only made with the 2 organisms.
That's a great question, Dr. Mariana. So in this one, we added 3 different cultures.
Okay, so after the yogurt was manufactured by the fermenting culture. Okay.
Yeah.
Fantastic presentation. I was wondering, have you looked into how the probiotics are associated with the genes that are expressed for quality in terms of fat content, protein content, phenotypic traits at all?
Yeah, that's a great question. So we did some follow-up study. We mainly looked into how these probiotics affect the qualities, water holding capacity, then some of the physical characteristics like color and also the pH. But we didn't look into too much details. Some of the basic yogurt characteristics.
Thank you.
Thank you.
Hi, this is Nurhan Danford. I'm just curious, in the stores, grocery stores, you can buy these yogurt-based probiotic enhanced products, right? Are they just adding those probiotics after making the yogurt, as you said you did that, right?
Yeah, that's a great question, Dr. Linford. Like one of the best example, if you buy yogurt from Walmart and Sprouts, Walmart, they add the basic starter culture. Whereas when you buy from the Sprout, you can look at it, you can see more probiotic cultures. So different companies add differently. I'm sure that will price in, that will affect the price too. So you can read the label and see which company adding, and some companies don't add.
Well, when they make the yogurt, I heard that they sterile or they heat process it, right? And then during that process, you lose those lactic acid bacteria and so on. So adding the probiotic afterwards. So I was not quite sure how that heat treatment process affects.
Yeah, again, I'm not a bio-expert by any means. My best guess, some of them are thermophilic bacteria, so they can survive the high temperature, especially Streptococcus. Yeah.
Okay, thank you.
And that's time.
Thank you, Chuck.
You're welcome. Thank you.
Thank you.
I guess this is weird. I have to introduce myself.
Ladies and gentlemen, Mr. or Dr. Peter Mariana.
Well, thank you. And to
also known as Microman,
coming at the trail end of this and just giving a kind of a summary of 2 overlapping projects that both are involved in related to natural nitrite. There's the isolation and characterization of nitrate reducing bacteria to generate the vegetable-derived natural nitrite, and then validation of its effectiveness against Clostridium spores in low and high-fat hot dogs. So as we heard from our keynote speaker, Jason, he presented a good background on that. It saves me from having to reiterate this, but again, sodium nitrate, nitrite are regulated food preservatives, and they must be listed on the label. That's usually a negative in turn for a processor. And then when it came to the category of natural meat and poultry product categories, USDA had to, they identified that you can't have any artificial food coloring or any artificial preservatives. So that created a dilemma. What do food processors do with sodium nitrite? And so USDA deemed, because vegetables have such high levels of nitrate, you can see here different vegetables and high levels of nitrate, and USDA indicated that consumer exposure to nitrite, which we often correlate with a bad thing, well, 95 to 98% of their exposure is from consumption of vegetables, and the vegetable nitrate gets converted to nitrite in the intestinal tract. So USDA said that, okay, if you use converted vegetable nitrate into nitrite, then that would be considered natural nitrite. And then you have the label claims that go with it, that they say you can claim no added preservatives and nitrate, no nitrates or nitrites added, or no artificial ingredients and so forth. And obviously that was a boon to processors who kind of like that idea to throw that on the label. And the only offset was that they can't call the product cured. So in our first project, we were looking at isolating bacteria that can do that fermentation modification of nitrate to nitrite. We took a broth assay that's typically you add two different reagents, A and B, sulfonylic acid and alpha naphthalene. And if you cultured your organism in a nitrate broth and you end up with nitrite, if you add those two reagents, you get a red color. If you don't get a red color, then it either means one of two things. It wasn't able to do that conversion. And you can add zinc powder. If you add zinc powder and get a red color, that'll confirm that you have nitrate and not the nitrite, because the zinc powder will reduce the nitrate to nitrite, and then the presence of those two reagents you previously added will then give you that color. If you do all that and you still don't get any color, then the answer, the alternative, is that your organism converted nitrate to nitrite and then further to other nitrogenous compounds. So we made an agar method that would facilitate screening nitrate reducing bacteria from food samples or from animal derived samples from our slaughter facility, the GI from their intestinal tract or the rumen. And you can see here, we basically made a base agar method where we sandwich the bacteria that's plated, sandwich it with a nitrate agar, let the colonies grow up. And then we have two subsequent overlays with reagent A and B. And we get some nice, pretty red zones around particular colonies that are nitrate reducing bacteria. And when we isolated them and identified them, there's different bacteria, some gram negatives and gram positives. And we're kind of excited about that because some of these bacteria are also known to produce bacteriocins that are perhaps antimicrobial peptides that are inhibitory to foodborne pathogens. So we might have the possibility in the future, when we go back to examine these, of not only having nitrate reduction, but also simultaneously making a bacteriocin as well. So we also quantified the nitrate and nitrite. And here's using an HPL ion pairing C8 reverse phase HPLC procedure. We were able to get some nice baseline levels of nitrite and nitrate standards. And we generate our standard curve, an excellent R squared correlation of 0.994 and 0.995. We also examined celery juice, which has nitrate. And when we fermented that with the industry standard organism, Staphylococcus carnosum, you can see nitrate was now converted to nitrite. And we also used one of our isolates, one of our better isolates, Staphylococcus capri, and also recovered nitrite from the nitrate that was there. So this is one part of our presentation. This was recently published. And our other one was to use natural nitrite. And that's where Jason Reicks' company, Florida Food Products, provided us the celery nitrite to use in these products. And we had to also to inhibit spore germination in low and high fat franks, and we used Clostridia sporogenes, which is a surrogate for Clostridia botulinum. and this BSO1. It's not a pathogen. So we used 3 different strains, harvested, compared harvest procedure for spores in both a broth assay method and a on-agar method of sporulation. Got equivalent levels from both, but we kind of like the in-broth, the broth method better. And we were able to recover a high concentration of spores. When we examined the spores by electron microscopy, you can see here the vegetative cells on the upper left after extended period of incubation in media, the spent media they don't like, and they sporulate. We get a high degree of sporulated cells, and under higher magnification, you can see the typical club-shaped terminal endospores of Clostridial spores, as opposed to perhaps the central endospores of Bacillus. So these are club-shaped appearance. When we went through our procedure for isolating the spores, we end up with a lot of spore coats that have spores inside of them. So the spores are contained inside the exosporium spore coat. We had occasional samples where we could see the individual spores, but for the most part, we recovered high level of, and pretty pure, of exosporia with the internal spores. And just some pictures I grabbed off the internet on the right, you can see the exosporia with the internal spores now germinating to have the protruding vegetative cell that's protruding. And that's what you want to use the nitrite to prevent that from happening in processed meats. So then we examined in hot dogs, in low and high fat hot dogs, we did duplicate replications, and we did triplicate samples at each sampling period for each replication. And we had 3 different batches overall. Control, where we had no nitrite, but spores added. We had sodium nitrite with erythorbate as the accelerant with spores added. And then we had celery, natural nitrite, celery nitrite with the natural analog to the erythorbate, the cherry powder or ascorbate plus spores. And you can see we had these made in our meat pilot plant, thanks to Jake Nelson and the crew. Arjun Busal here was doing some of this work for his degree. And also you can see the product at the end, no nitrite, you get that brown color. Sodium nitrite or celery nitrite with the appropriate accelerant gave that nice red color that you expect of hot dogs. And so we, again, we had low and high fat hot dogs. We had different portions of that batch held at 5 degrees centigrade, 15 degrees centigrade, and a more permissive 35 degrees centigrade. And I'll just jump to the conclusions here that, or the results, that even under accelerated shelf life conditions of 15 degrees centigrade, we got no move off baseline for up to 20 weeks on some of these trials. This one didn't run that long, 8 weeks, but most of them went 18 to 20 weeks. And it was only in the permissive conditions, 35 centigrade, which is 95 Fahrenheit, did we get immediate growth out of the controls where there's no nitrite. And you can see the samples with the nitrite, we basically held off any kind of growth for up to 2 days, and 3 to 4 days, we started getting some growth there. So you have good control for 2 to 3, 1 to 2 days, definitely, at even 95 Fahrenheit. So our conclusion from this is that we had no significant difference between sodium nitrite and celery nitrite. You can see here, they're pretty much the same, okay, all the way through on all these different trials. No significant difference in effect in low versus high fat hot dogs, pretty much the same here as we have down here. And also, the thing I wanted to bring up was that Jason was bringing up the point about Appendix B cooling. And we often get questions from processors, our cooling procedure, we went beyond the time frame that we didn't get chilled in time. We went about an hour or 2 longer. Is our product okay? And in the past, not having worked with this directly, I would say, well, what you went past the time period, I guess you can take a gamble if you want, but I wouldn't. And then looking at this data, that even at 15 degrees centigrade for weeks, nothing happened. And here at 95 Fahrenheit for 1 to 2 days. So I'm almost tempted to say there ought to be some kind of revisiting of those conditions when you bypass the temperature hold conditions for just a short amount of time. And so with that, I'd like to acknowledge our Foundation of Meat and Poultry Research, the Beef Checkoff, our USDA NIFA National Needs Fellowship funding, the Advanced Foods Gilliland Professorship, Microbial Food Safety, and also Flora Food Products for providing the celery nitrite. And with that, I'll answer any questions that you may have.
2 minutes for questions.
Oh, wow. Okay.
Pete, one of those pictures, the celery nitrate had a plumper hot dog compared to the no nitrate and the traditional. Is there a reason why that one came out?
I guess that was one of the funky ones. They, you know, you got to tell students take the better ones to take a picture of. No, I guess that was, you know, I guess they're not. Some
I
of
thought
the
there
hot
might be
dogs,
a marketing advantage. It's plumper and juicy.
No, I don't think it had anything to do with it. I think it was just the one that they chose, you know, to use. You can see some of them here. I mean, this one might have missed the linkage. It's like twice as long, so that's like a foot long dog. So, I don't know. Maybe there is something. We'll have to check that out. A plumper dog. Yeah. Any other questions? Well, again, some of the things that I was interested in the future is to evaluate the use of bringing in bacteriocins to either complement or maybe replace some of the nitrite activity if it's inhibitory to Clostridia. And so whether we can have the same bacteria that do the modification of nitrate to nitrite, ferment the nitrate to nitrite, also making bacteriocins, you might get two complementary things in that same fermentation, or simply add external bacteriocin from another organism fermented with another organism. Also, to compare the effect of vegetable nitrite versus sodium nitrite on perhaps a non-toxigenic isolate of Clostridia botulinum and perfringens. And also, I didn't bring this up, the HPLC analysis worked great in broth. When we tried to do the same thing on samples from the hot dogs, we weren't able to get a nice clean assay on the HPLC. And I think it had to do with all the meat components complexing the nitrite and nitrate.
Hey, Pete, Mark Vaughn, has there been any work done on any effect on E. coli as far as is reducing that prevalence? I mean, I see you've done work with Listeria, and of course, botulinum would be a different animal altogether, but any work on Shiga producing toxins?
Well, I've done work with the bacteriocins against Listeria on hot dogs, and we've we had some pretty good results. I can send you that work if you're interested in. And basically using a cultured fermentate of some of our bacteriocin producing strains,
right,
that inhibited Listeria. So and the nice thing about the bacteriocins is that they're heat stable, they're small peptides, there's not much tertiary structure to denature, and so they'll tolerate a cook process easily. But the problem is generally for like Listeria or other organisms, except for the spores. Spores are likely to be internal in the meat matrix, but for a lot of surface contaminants like Listeria, it's usually an external contaminant, and you could just spray it on the product and get pretty good inhibition. But you were asking about E. coli, I suppose?
Yeah, I just was wondering, I grow lots of celery, but, I don't process it in this, but, could this somehow have an impact? Could this be, could we get some reduction at the field level by including something like this in irrigation water? I don't know. Just came to
so-
mind. I know this isn't what you studied here, but I was just,
Well, maybe we can get back if there's some interest, we can maybe pursue some type of project in that area.
okay.
I have a comment. This is fantastic symposium. I appreciate being here and listening to you all speak. As far as the nitrites and nitrates, I'm interested in that process because you're familiar that happens in aquaponic systems at all? Are you familiar that the conversion of nitrites to nitrates there in the aquaponic system at all?
No, I'm not.
Okay. I was just wondering if you might be interested in that conversion and trying to identify what might be going on in that conversion from nitrites to nitrates in that system. It might be interesting to correlate what goes on there and what you're trying to do in the system that you're working
Oh,
on.
what's the, I'm trying to see what the, how that would work.
From ammonia
Oh, okay.
to nitrites, then to nitrates by bacteria or by microorganisms.
Going the other way. Yeah, going the other way. Okay.
Yeah, so.
Okay, interesting. Okay. With that, if there's no other questions. Chuck?
I'm trying to, there I am. Okay. Yes, so wonderful presentations. And on behalf of the Institute of Food Technologists Oklahoma section, I want to commend all of those students that presented today. Did a really fine job. IFTOK is, of course, our co-host, co-sponsor for this event and has been for many years. I also want to thank Stillwater Centennial Rotary for their financial support and also the support from the Oklahoma section for the International Association of Food Protection. And so I think everybody's wanting to know, well, how'd we do? Who gets to take the money home? So I've got that right here. Let's start with the graduate student category.
Hey, Chuck,
Yes.
could we ask Pete to stop his screen sharing real quick before
Oh,
you?
still there?
Yes.
Yeah, you're still, well,
How do I do, how do I,
Down at the bottom, it should say stop sharing.
hold on. Where am
All
I?
right, here we go.
Oh, here it is. Stop share. Okay.
Okay, thank you very much.
Sorry about that.
All right, yeah, I don't have a certificate to share on the screen. Those haven't been printed yet, but the certificates will be ready. If you're on campus, you can pick it up or you can wait for it to go in the mail. An email will go out to all the winners as to the certificates and their winnings.
I think everybody is a winner for presenting. I mean, they did a great job. They took time out of their, not out of their day. They must have worked on this for a while as well in the abstracts and submitting them. So thank you everybody for participating. And I can't wait till we get back to the in-person research symposium next year.
I put the hand up there to clap too, so, because that's Zoom etiquette. All right, now without further ado, let me announce third place graduate students. We have a tie. So each one of these awardees will be awarded full 150. You do not have to split it. So $150, third place tie, reduced lignin, alfalfa, and water limited environments, Elena Gerhart. Elena, go ahead and turn on your screen and wave. That's as best we'll do as a handshake.
Yeah.
Okay, I see her. She's square number 7 on my screen. We'll move right along. And our other third place winner, tying and receiving $150. Dark storage of enhanced dark cutting beef in nitrite embedded packaging, increased metamyoglobin formation upon repackaging. Morgan Denzer. Morgan, wave hello. Very good. Good job. Now moving on, second place and winner of $300 in the graduate student category. Student views of insect consumption, AQ methodology study. Melissa Reed. Melissa, congratulations. All right. And finally, first place, winner of $500 in the graduate student category, use of alternative salts in Biltong marinade to reduce sodium and still achieve USDA FSIS greater than 5 log reduction of salmonella, Caitlin Karolinko.
Oh,
All right, Caitlin, good job. Congratulations. Now let's move along to our undergraduate students, and I commend all 6 of you for a really good job. Well done. One third place winner, $150. Use of brewer's spent grain for production of high value mushrooms, Lauren Millis. Very good. And now for our second place, winner of $300, prevalence of selected pathogens and ectoparasites from feral cats of Central Oklahoma, Kristen McKay. Good job, Kristen. And finally, at first place, $500, designing a knockbox for the smaller packing facility, one structure for bison, cattle, swine, sheep, and goats. Sydney, Cannon, and team.
yeah,
Unfortunately,
that was a team presentation.
That was a team
We haven't had one of
presentation.
those before.
That was very unique. First time we've ever had it. I will not be writing 3 $500 checks, however. So, but very good. And I guess with that, I would like to turn this back over to Peter Mariana. Once again, thank you for our sponsors for providing the award funding for this and the support so that this can continue, whether ICE or COVID. Pete, back to you.
Well, I mean, I wanted to make a comment. I think it was based on who had the mushroom one, Lauren. Laura and I used mushroom compost in my garden one time, and now I've covered that up and made lawn. And now I've got mushrooms coming up all over the place every year. I'm out there kicking them like left and right every morning. You know, about 40 or 50, I get tired of it. So I don't know how you stop them, but it's driving me crazy. But just to again, congratulate everybody for doing the presentations. Congratulating everyone else for participating and listening to all these. And again, thank you. Can't wait till next year we have it in person. I miss those workshop luncheons too, you know. So so anything else? Dr. Escobar, I notice he's here. Have any words to say to our guests here that are here?
Thank you, Dr. Mariana. Just a great job of all the presentations. They were really, really amazing. I enjoyed all of them. And I think they were better than the 2 faculty members' presentations.
Well, there wasn't any money in it for it. I did it quick, so.
And our guest speaker, thank you for a keynote. Really appreciate it.
All right, well, with that, I guess call it a day here, or we adjourn? We have a motion to adjourn.
I second.
Okay.
Adios, amigos.
Take care. Thank you all.